So welcome back, everyone, after this fantastic second keynote. Welcome to a session on sensory systems. As you've all probably figured out by now, you can use the chat window to talk about the talk, but if you want to ask questions, please push the Ask a Question button and ask a question there. So, Anton, if you can put your slides on now. Okay, lots of echo, sorry, I'll put in. Is it better now? Okay, does it work now? Can you still hear me without echo? Okay, great. Thank you. Okay, so our first speaker will be Anton Arkhipov from the Allen Brain Institute, and he'll talk about towards multipurpose biorealistic models of cortical circuits. Anton, the floor is yours.
Okay, thank you. So can everyone hear me? Let me just double check. All right. Okay, I'll go ahead. So yeah, hello everyone. Thank you very much to the organizers for letting me speak today and for putting together this really nice conference. I'm coming from the Allen Institute for Brain Science in Seattle, which is sort of famous for providing large, complex, and high-quality data sets on different aspects of brain composition, connectivity, and function. And today I'll be telling you about how we use this data to build models, biologically realistic models of cortical circuits, currently with the focus on the mouse visual cortex. So these are the models that we constructed. They've been published recently, so most recently in this paper in Neuron. And they, as I said, focus on mouse V1, primary visual cortex of the mouse, so here at the back of the brain. This is the illustration. So they span, these models, they span all 6 layers of the cortex from layer 1 down to layer 6. They contain 230,000 cells approximately, incorporate a lot of the realistic biological data, include multiple cell types, cell classes, so 17 cell classes in this particular case. And we built 2 versions of these models. So one is a biophysically detailed model where the central portion of the model is represented using biophysically detailed compartmental morphologically realistic neuronal models. And the other, on the right here, is a point neuron version, where each neuron on the left, the biophysically detailed model of a neuron, is instead now represented by a point neuron, generalized leak integrate and fire models. And so there is a one-to-one mapping between these biophysical and point neuron models in the sense that every single neuron, its coordinates, the connectivity to all the other neurons are exactly the same between these 2 models. And so basically they are copies of each other, with the only difference being that the level of resolution that's being used to represent neurons in one and the other case. And so if there is only one thing that you can take from my talk today, I would urge you to remember this, that these models are freely available. We've built them, we optimized them, we did some science with them and published them in papers, but the main thing is that we made them publicly available to everyone here on this website. You can download them together with all the metadata that went into building these models, simulation results. and you can use them for your own purposes. Of course, the biphysically detailed model is quite computationally expensive, so you need hundreds of CPUs to run it efficiently. You can get maybe 1 simulated second in 1 hour of computational time. But the point neuron model is certainly suitable for almost any lab. So it runs on a single CPU. You get about 1 simulated second in 3 minutes. And so, yeah, we really welcome people using these models. If you are working on the visual system, visual cortex, great. If you don't, maybe you can consider this as a sort of a zero-order approximation of a cortical canonical circuit and use it for whatever purposes, make perturbations, test your theories, make predictions. So it's all there for everyone. Okay, so how did we achieve this? We've built some tools, and I'll just mention here that we present these tools in a workshop on Tuesday, the workshop on tools and resources for developing and sharing models in computational neuroscience. There will be a lot of very interesting talks there about different tools, including ours. And so the tools are the Brain Modeling Toolkit, which is a software we built for running simulations. It doesn't run simulation itself. It's actually an interface to establish tools like Neuron and Nest, but it allows one to interact with these tools quite efficiently and conveniently. And then another thing we created is this modeling file format we call SONATA. We developed it together with the Blue Brain and other colleagues. And so this is a format to represent models efficiently. And that's how we share our models with the community. So both tools are there, and come listen at the workshop if you want to hear more. Okay, and so also how we build models, I'll discuss in detail in another workshop W6 here on open data tools and models from the Allen Institute on Wednesday. And this is going to be a really cool workshop where I will talk about all kinds of different aspects of the Allen Institute data, as well as how this data are being put together into models. But briefly, basically what's happening is that at the Allen Institute, we have different types of data. For example, data that characterize morphologies and electrophysiological properties of individual cells. And so we combine this for different cell types into models of single neurons and then put them together into this model of the whole circuit of the whole cortical area. There is data on the connectivity, connectivity between individual neurons and different neuronal types. And actually for this work, we mostly use data from the literature, but in the recent couple of years, a lot of really cool new data came out at the Allen Institute, and they're really ripe for developing next version of the model. And then finally, neurophysiology in vivo. And so primarily we've been using neuropixels recordings, so large-scale electrophysiological recordings in the mouse visual cortex and thalamus in vivo. And so these data are used to first optimize models to make sure that what they do is realistic and in the sense of the firing rates, the distributions of firing rates. But then certainly they also used to test the models. Once these models are constructed and optimized, we test them on the held out data to see how well we perform in comparison with the experiment. And one thing I'll mention quickly here is that the optimization, usually we do the optimization on a very small subset of the data, very, very specific stimulus, so a particular instance of a drifting grating, but then we test the performance on the model on a variety of different stimuli and different metrics. And so let's talk about that a little bit here. So one other aspect of this model is that we've built a relatively realistic representation of visual inputs for this model. So that's done using over 17,000 models of thalamic cells of 14 different types, all of which were tuned to reproduce experimental measurements in vivo. And those cells are represented as filters that operate in the visual space. And so because of that, one can use arbitrary visual stimuli, drifting gratings, static gratings, noise, natural scenes, natural movies, anything that, let's say, your experimental collaborator is using to provide visual stimuli for their experiments. You can use, take it, run through our model, any movie, and of course it will take some computation, but after that you will have spike trains that can be used to supply inputs to our model. And the way how those spike trains converge on individual neurons in V1 is really, we spent a lot of time to constrain that using a lot of experimental data from the literature. The bottom line here is that arbitrary visual stimuli can be processed. And as a result, we try to run our model with a number of different visual stimuli and then compare with the experiment. So this is one example. Here on the right, we ran simulations where we turn on a grating stimulus at 500 milliseconds and then run the simulation for 3000 milliseconds, showing the activity here of 50,000 neurons in the center. So a lot of different patterns of activity for different neuronal types. And then we can take activity of these types and compute distributions of different characteristics. So in our paper, you can see that we computed a lot of different metrics for different stimuli. And one example here is the direction selectivity index. So how directionally selective neurons are. We can compare for different cell types of the experiment. So in gray here, and then two versions, the biophysical and the point neuron version of the models. And you see that for this metric, the model does relatively well. For some other metrics, some metrics do well, some do less well, but overall, we found the performance to be pretty good. So this is testing the model, and then you can ask for the metrics that show less good performance. Maybe these are the directions in which the improvement should be made in the future generations of this model. But then we also, in doing this exercise, we've came up with some predictions. And this is, again, just one example. You can read more in the paper. But one thing that we found has to do with this like-to-like connection strength. And so that basically works this way. If you have two neurons that prefer similar stimulus, let's say the same orientation or similar orientation of a moving stimuli on the visual field, the connectivity between them, the strength of the connection is stronger than when the neurons prefer different stimuli, especially orthogonal ones. So that's what we refer to as a like-to-like connection strength. And we found that was really important to capture experimental observations with our model, in particular direction selectivity. And it was especially important for certain cell types. For example, for interneurons of SST and VIP types, turns out they do not receive much of the external inputs from the thalamus. And so the recurrent connections with this like-to-like properties turned out to be really important for them. And so here we do a simulation where in orange is our original model, and you see that different layers in the SST interneurons have relatively high direction selectivity. But then when we change the connectivity rule and now the connection strength is uniform, then the direction selectivity of those neurons becomes very small. Okay, so finally, now that this model is ready, we offer it to the community, but we also continue doing work on several projects with this model, trying different applications. And one application that I want to highlight is perturbations. So of course, this is something very sort of hot area in experiments. And with the models, you have a lot of control here. And you can explore a lot of different parameters. So just an example, this is an intact model running a simulation for 3 seconds, but then if we silence excitatory cells in layer 4, then you get this. Of course, layer 4 is completely silent, but then other neurons are strongly affected as well. And we can do that for any cell of any type the way we want in the model. So in this project, we are working together with internal collaborators, Stefan Michalas and Bin Huan Cai. and also with external collaborators, so with Chris Harvey at Harvard. So Chris published this paper last year where he showed that perturbing a single layer to 3 neuron produces this sort of center surround effect where other neurons are excited. So if we excite a layer to 3 neuron, right, the other neurons that are close to it are also excited, but neurons further away are inhibited. And we observed the same thing. So that's good. That's a confirmation that our model works well in this respect. Look at further. And so this is an example of trying to make a prediction now with the model, which is reported in this bioRxiv paper, where we basically show that the system can switch between the efficient and robust coding. So here we excite a certain number of neurons that are similarly tuned from 1 to 100. And then in these orange and blue lines, look at what happens with similarly tuned neurons. And it turns out that for high contrasts, for strong stimulus, these similarly tuned neurons, their activity is suppressed. So that's efficient coding, right? If you have a lot of neurons that talk about the same thing, a few of them start talking, and then the rest can shut down and save their effort. So like efficient coding. But then if the contrast is 5%, so there's a small weak stimulus, turns out the trend is different, and now instead the activity of the similarly tuned neurons goes up with excitation of more and more neurons. And so now this is more like a robust coding where you have all neurons talking and supporting each other. And so what we show here is this model, and it looks like we can switch between these two different regimes. Of course, needs to be tested experimentally, but one example of what types of studies this model can be used for. And there are many more. I just don't have time to talk about all of them. But again, so I'll finish here. Basically, we have these models. They are open for the community. Please use them and give us feedback. And then finally, I invite everyone to join this workshop that we will do in a month from now, where we will talk a lot about models, the Allen Institute models, other models, a lot of exciting speakers, experimentalists and modelers. And so please join us there. Okay, and thank you for your attention.
Thank you very much, Anton. Okay, so there's one question here. So in the interest of time, I'll just read the question. So to what extent could be your V1 connectivity used as a proxy for a canonical neocortical circuit or column? And how much is it different from other neocortical areas?
Oh, that's a great question. Well, so I think to what extent depends really on your purposes and applications. And also how is it different from other cortical areas? I mean, to a large extent, we don't know. V1 is one of the best studied cortical areas in terms of understanding its connectivity. We know that some other cortical areas have substantial differences in composition or connectivity. For example, V1 is at the back of the brain. If you go to the front of the brain, you will have, for example, fewer PV inhibitor neurons. In some cortical areas, you essentially don't have layer 4, which is very important, input layer in primary sensory areas. So, ultimately, we don't really know for a lot of the areas. We just don't know at the micro level how the connectivity is organized. And frankly speaking, we don't really know that for V1 as well. There are a lot of data here and there. In building our model, we had to use some data that were not from V1. We had to use some data that were not from the mouse even. But I think especially for sensory area, that's a good first order approximation. There are some similarities, and you probably can use that more.
Okay, thank you. And maybe a second question. So how much do you lose in the point neuron model version with respect to the biophysically detailed version? And which type of experiments are you not able to reproduce with the simplified version, if any?
Yeah, excellent. Well, so I personally think that the point neuron version is is good to use for a lot of applications. We are using right now, once we build these models and compare them extensively, I would say we probably use point neuron models, I don't know, like 70% of the cases. But it certainly depends on the applications. We found that for the firing rate characteristics that we tested, the two models perform very similarly. But there are some questions that you simply cannot address with point neuron models, or at least it's very difficult. So one example, a project that we have that we are working on together with Gaute Einevold in Norway is computing local field potential. And in principle, you could do it with point neuron models, but biophysically detailed models are more amenable to that. So yeah, it should be really driven by a question you want to ask.
Okay, thank you very much. Okay, so Anton, I'll kick you out. Thank you again. And I'll invite the next speaker on. Yes, and for more questions, there will be on Eurostars, you'll find a dedicated page for your talk, and then you can ask your questions there, and Anton, you can answer them. Thank you.
Yeah.
Hello.
Okay, hello. Hi. So can you try to share your screen?
Sure.
Okay, great.
Okay.
So our next speaker is Ali Almazi from the National Vision Research Institute in Melbourne, and he'll talk about how similar statistics affect receptor fields of neuron cells.
Thank you very much for the introduction. So I want to talk to you about how stimulus statistics change the receptive fields of cells in primary visual cortex. So a great deal of what we know about the sensory coding in the visual system is based on a stimulus response characterization of neurons. For example, to study the kind of processing that happens in the primary visual cortex, we often present a bunch of visual stimuli to an animal, and we record the neural responses to those visual stimuli, and we treat the whole system as a black box, and we use some system identification techniques to see how the output of the system, which is the neural responses, depend on the input to the system, which is the visual stimulus. So this characterization of a neuron based on a stimulus response gives a description for this neuron, which is known as its receptive field. The question that we asked was that how much this characterization depends on the type of stimulus that is presented to the neuron. Or to be more specific, if we present two completely different set of stimuli like white noise and natural scenes to the neurons in primary visual cortex, what would be the difference in the way that these neurons respond to these two stimulus types? So to characterize the receptive field of a neuron, a model is often used, and this is the model that we have used in our study. So in this model, these filters account for the receptive filters of the cell, and these bands of red and blue indicate the on and off subregions of the filter, which are sensitive to increments and decrements in light intensity. And the output of each filter indicates the similarity between the visual input and the spatial structure of the filter. It basically tells us how much of this spatial structure or feature is embedded in the visual input in terms of the contrast of this feature. And because of this, we call the output of each filter its feature contrast. And in the next stage of the model, there are some nonlinear functions called input functions that are applied on the output of these spatial filters, and there are some up to form a generator signal for the neuron. And there is a spiking nonlinearity in the in the form of a parametric group exponential function that converts this generated signal into the firing rate for the neuron. So this model has a great advantage that it does not assume any particular shape or form for these spatial filters or these nonlinear functions, and they are estimated non-parametrically from the neural data, which makes this model also biologically motivated. So again, the aim of our study was to see how characterization of V1 cells using this model change as the visual input changes between natural scenes and white Gaussian noise. So we did a series of experiments in which we recorded from the cat primary visual cortex using multi-electrode linear probes, while at the same time we were presenting interleaved blocks of white Gaussian noise and natural scenes to the animal. So these are examples of the stimuli that we've used in our experiments. So on the left there is an example of a natural scene, and on the right there is an example of white Gaussian noise. These two stimulus types were matched in mean luminance and global RMS contrast, but the main difference between these two stimuli was in terms of the second and higher order statistical dependencies, which are very strong in natural scenes, but they don't exist in white noise. And to characterize or uncover the receptivity of a neuron, we basically estimated the parameters of the model that I showed you before using a maximum likelihood estimation method, which was done on the set of all model parameters simultaneously using a joint optimization. So this is an example of the way that a V1 cell responded to these stimulus types. So what I'm showing here is the post-stimulus time histogram of the responses of this cell to natural scenes in green and to white Gaussian noise in magenta. So we can see there are pronounced differences in terms of firing rate and response latencies between these two response patterns. So this cell responds with a higher firing rate and shorter latency to natural scenes than to white Gaussian noise. We also looked at the whole population of the V1 cells, and for each cell we plotted the response latency to natural scenes against the response latency to white Gaussian noise, and we did the same thing for the firing rate of the neurons. For the response latency, we noticed that all cells responded with a shorter latency to natural scenes, which was on average about 11 milliseconds shorter than white Gaussian noise. And most V1 cells responded with a higher spike to natural scenes than to white Gaussian noise images. So now I'm going to show you an example of a receptivity characterization of a V1 cell using natural scenes and white noise. So this is a cartoon image of our example cell. When we presented natural scene to this cell, its response was best described using a receptivity structure that had these three spatial filters. But when we presented white Gaussian noise to the very same cell, its response was best described using this receptivity structure. So the most obvious difference between these two receptivity structures is in terms of the number of spatial filters. So there are more filters in the receptive field characterized using natural scenes, which makes the receptive field more complex, and it means the cell responds to a more complex set of features in the stimulus. So we then looked at the distribution of the number of spatial filters for cells in our population data using natural scenes in green bars and using white Gaussian noise in magenta bars, and we noticed that there are indeed more filters for the characterization using natural scenes. We also looked at the distribution of the difference between the number of filters for the receptive fields characterized using natural scenes and white Gaussian noise for the same cell. And as it can be seen, this distribution is very asymmetric, and it is skewed to the right towards having more spatial filters for the characterization using natural scenes. So we looked for reasons to explain our findings. So first we had to check if the extra receptive filters that we get using natural scenes are not artifactual, or because of any other issues, for example, because cells often responded with higher spike rates to natural scenes. So since we've used maximum likelihood estimation, we can make sure that there is no artifact in the uncovered receptive filters. Also, we did a control analysis, and it turned out that identifying more receptive filters on natural scenes is not actually related to the higher spot counts of V1 cells on these images. So this cannot be an explanation for our findings. The other explanation that we thought of was adaptation of V1 cells to the statistics of natural scenes and white Gaussian noise. So adaptation can be defined as a change in the model to better describe the cell responses under a new stimulus regime. So it is model dependent in this case and can be captured using the mathematical description that we have for a cell. So if I go back to the model, a change in this model can be either as a change in the receptive filters or a change in the receptive nonlinearities, including the input functions or a spike in nonlinearity. So far, I've only talked about the changes in the number of receptive filters, but another thing about these filters is that they have significantly different output distributions on natural scenes and white Gaussian noise. So I mentioned that the difference between the white noise and natural scenes is in their second and higher order statistics that mostly account for features like edges and contours in natural scenes, which show a strong similarity to the kind of receptive filters that we find for V1 cells. So this means that the distribution of the output of these filters on natural scenes would be very different from white Gaussian noise. So take this filter for example. So if I apply this filter on our natural scenes and white Gaussian noise images, these are the distributions that I get for the output or feature contrast of this filter on natural scenes in green and white Gaussian noise images in magenta. So note the difference between the broadness of these two distributions. We get a much broader range of feature contrast on natural scenes than on white Gaussian noise images for these V1 receptive filters. So it is possible that V1 cells adapt their dynamic range to account for this significant change between white noise and natural scenes. So to see if this is actually the case, we did a separate control analysis in which for each cell we matched the range of feature contrast on natural scenes to the range of feature contrast on white Gaussian noise, and we estimated the number of receptive filters for cells under this matched feature contrast natural scenes. So for example, this corresponds to the range of feature contrast of this filter on white Gaussian noise images. Now, if I only sample natural scene images that have a feature contrast within this range and use them for receptive field characterization, I can match the range of feature contrast of natural scenes to the range of feature contrast of white Gaussian noise for this filter. This is the distribution of the difference between the number of filters for natural scenes and white Gaussian noise that we had seen before. After matching the range of feature contrast of natural scenes to white Gaussian noise for these cells, and we estimated the number of receptive filters, we get this distribution. So we can see that for most cells, the number of receptive filters found using this matched feature contrast natural scenes has dropped to the number of receptive filters that we have found for these cells using white Gaussian noise images. And in other cases, the number of identified filters using this matched feature contrast natural scenes was either less than white Gaussian noise or no filter was identified at all. So this suggests that the higher range of feature contrast for V1 cells on natural scenes is actually responsible for finding more receptive filters for these cells on these images. And also when presented with natural scene images, V1 cells operate at a different regime than when presented with white Gaussian noise, and different dynamics of neural processing might be in place. We also found significant differences between the receptive and nonlinearities of V1 cells between white noise and natural scenes. So this is an example cell, and for this cell we found a single filter using white Gaussian noise characterization, and then single filter using natural scene characterization. And the difference between these two filters was statistically insignificant. For the response nonlinearities, what I'm showing here is the composition of the input function and the spike in nonlinearity in the model, which directly relates the output of these filters, which is the feature contrast, to the response of the cell in terms of firing rate. And that's given for the characterization using white noise in magenta and natural scenes in green. So first, note the difference between the domains of these two functions, and that's because of the difference in the range of feature contrast of these two filters on white noise and natural scenes. And second, see how the natural scene response function has shifted to the right, similar to the contrast gain control effect that we often see for V1 cells. We can also compare the response functions within this shared domain, which corresponds to the range of feature contrast for white gas and noise images. And if we do that, we can see that when the stimulus changes from natural scenes to white noise, there is a significant amplification in the response function within this low feature contrast range corresponding to the feature contrast of white gas and noise images. So we generalize this analysis to cells with different number of filters on white noise and natural scenes, and we use a quantitative index to measure these changes in the response functions between these two stimulus regimes. And we found that for 85% of cells, when the stimulus was changed from natural scenes to white noise, there was a significant amplification in the response function within the range of feature contrast of white gas and noise images, similar to the pattern shown in this picture. And for about 5% of cells, we found the opposite effect. And for about 10% of cells, we found no significant changes between the response functions of the two stimulus regimes. Now, taken together and to wrap up, the cells that showed an adaptation effect in our population of V1 data as a result of the change in the stimulus statistics were about 90%, which were in the forms of a possible change in the number of receptive filters and a change in the feature contrast response functions. And among these cells, more than 95% of them showed a significant amplification in their response functions within the low feature contrast range when the stimulus was switched from natural scenes to white gas and noise. So I would like to finish by thanking all the people that were involved in this study, and thank you very much for your attention.
Thank you very much, Ali. We have time for one, maybe two questions. So the first question is, did you consider using phase randomized natural images as a third option, which would retain the spatial frequency and orientation statistics?
No, we haven't used any other stimuli, although we have used white and natural images, but that was just in one experiment. And so we found similar effects to natural scenes, because when we whiten natural scenes, we just remove the second order dependencies, but there are high order dependencies, high order statistics, and we see that we see similar effects to natural scenes, but we've done that for only like a handful of cells, so we don't have a population to compare them to this population that are presented in this talk.
Okay, thank you. And second and last question, have you looked at pairwise interactions? In other words, how is white Gaussian noise versus natural stimuli reflected at the coupling level?
So does that mean pairwise correlation between cells, or you mean between pixels in the
So maybe
images?
to specify more, if you wanted to decode white Gaussian noise and natural stimuli, do you think couplings would contribute to the accuracy of decoding, or should firing rate be enough?
Oh, so you mean like a population coding instead of single cell coding?
Yeah.
No, we haven't done that, because so the model that we've used, it's only for single cell encoding, and no, so we haven't looked into pairwise correlation of the neurons, although that's a good option to look into.
Okay, then it's third and last question just popped up, so I think we still have time for that. So do models estimated from natural stimuli do a better job of predicting responses to white noise than vice versa?
No, because there is adaptation, so the model that we get using white noise is best for predicting the responses to white noise, and the model that we have estimated using natural scenes is best to predict the responses to natural scenes, and that's another indication of adaptation between these two stimulus regimes.
Okay, thank you very much. Yes, so I'll kick you out, Ali, and then I'll invite the next speaker on. Thank you very much again.
Thank you very much, too.
So, welcome, Yoram.
Thank you.
If you could try to share your screen. Yeah, there we go. Great. Thank you.
Okay, can you?
We can see the slides, and now we can see your.
1 second, though. I want to. Well, you can see me. Everything is good because I cannot.
So we can see you. We can see your slides, but once you go full screen mode, we'll see your mode instead of the standard mode.
Well, now it's fine. Can I go on? Or you want me to go full screen mode? Because
if you can try once again, going full screen mode. If not, we can just go through it and present it or in slide.
No, I do want to go full screen mode for sure.
Do you have two screens?
I do have two screens. Yeah.
Maybe you're sharing.
No, okay. Let me. Sorry about that. It worked.
No worries.
I want to stop sharing, and I'll start sharing the other screen.
Okay. Yeah. Let's try that.
Oh, stop sharing here. Okay, and then I will share this screen, and I will share the screen one. Share. And
okay, we can see your screen now. Now we are in full screen mode. Perfect.
Okay.
Yeah. So our next speaker is Yoram Benchal from Hebrew University, and he'll talk about analysis and modeling of response features of accessory olfactory bulb neurons.
Right. So thank you very much. I should have been now in Melbourne, enjoying this conference and also visiting my sister, but unfortunately this is not happening. But I'm still happy to talk to you from where I am in Jerusalem. The original title was Analysis and Modeling of Response Features of Accessory Olfactory Bulb Neurons, but this is a big story, so I decided to focus on a small part of this. And so the more accurate title would be Stereotyped Presentations in a Randomly Wired System. So I have to explain the system that we study. It's a chemosensory system, as you may know, there are multiple chemosensory systems in many animals. The system that we have is called the main olfactory system, but there are others. And one of the most important systems, and the one that we study, is called the vomeronasal system. It's also known as the accessory olfactory system. Here you see a diagram in a mouse head showing the key structures of the system. And except humans and some apes, most vertebrates possess the system, definitely many mammals, including mice. And the system is, the way we see it, is largely designed for processing cues from other organisms. So mainly conspecifics, for example, other mice, but also from predators. But generally, it's a system that is supposed to process signals with innate meanings, such as pheromones. This is one class of signals. And here's a general layout of the vomeronasal system. So there are sensory neurons in the nasal cavity in a special organ. They project to structures that are called glomeruli in the olfactory bulb. And specifically, they project to a region called the accessory olfactory bulb. It's a small region within the entire olfactory bulb. And in the olfactory bulb, you have principal neurons, which are called mitral tufted cells, and they sample the inputs from these glomeruli, and they send their axons to regions in the amygdala and the hypothalamus that are known to control social and other innate behaviors. But unlike the main olfactory system, the sensory neurons of the vomeronasal system, they project to multiple glomeruli. And in addition, the mitral cells in the system, the projection neurons, they sample from a number of glomeruli, and it's a variable number of glomeruli. So here you see a very nice image by Ramon y Cajal showing you a section of the brain, and you see the AOB, and you see the part of the main olfactory bulb, the main olfactory system counterpart of the structure. And if you look at this, the AOB seems messy, and at least to me by eye, looking at several images, not just this one, the connectivity in the AOB seems random. And the question is, how can a system that's designed to process cues with innate relevance, which implies stereotyped responses, can be so random? In other words, can one achieve stereotyped representations despite this apparent randomness? So in order to answer that, I have first to explain what we did, I have to explain our data. So first of all, we have electrophysiological data. And in our experiments, what we do, we use multi-site electrodes, and we record activity of the neurons that I mentioned, the mitral tufted cells of the accessory olfactory bulb. We present stimuli to these to the sensory neurons, and then we measure the rate changes in the mitral tufted cells. So here are examples of two neurons. One is specific for female urine, the other one is specific for male urine. This is just an example to show you the type of data that we have. And the analysis that I'm doing now, each response is condensed to a single value, which is just the average firing rate over a prolonged temporal window. In our data set, we have about 240 AOB neurons. And importantly, we have responses to 6 different natural stimuli. These are urine mixes from males and females from 3 different mouse strains. Okay, so 6 different stimuli. In addition, we also have chemical data. So the chemical data are measurements of molecule levels for the very same stimuli that we use in our electrophysiological experiments. And we have 2 data sets. One is for volatiles, and the other one is for peptides. And the focus here is on peptides because these seem to be the more interesting class of molecules for these neurons. So just to show you an overview of the data, so this is a matrix showing you the responses of our of the neurons. So each column here corresponds to one of the 6 different stimuli, the female urine stimuli, the male urine stimuli, the different strains, and each row therefore is one neuron. And we see that the firing rate changes. And this is a similar description of the chemical data, of the peptide data. So again, the stimuli in here, instead of neurons, each of the rows corresponds to one of the different molecules. And so the experiment or the analysis that we want to do is that we want to simulate the responses of the neurons to these chemicals. I will explain how we do this. The parameters of interest that we want to explore are the number of mitral tufted cells. So in this cartoon, you can see 3 mitral cells that sample, each of them samples one of the molecules. But we also want to play with the number of chemicals that are sampled by each of the mitral cells. So here you see, again, 3 mitral cells. Each of them samples 5 different molecules. And here is the modeling. It's a very simple modeling, especially for this kind of a conference. But the response is a linear summation. It's a graded response. So there's no thresholding here. This is just a response of a neuron with a certain to certain stimulus is given by the neuron's tuning function and to the different molecules, and also the concentrations of the molecules in the specific stimulus. So we have 6 different stimuli, and so we can show this graphically as we have a tuning matrix of the neurons for each of the molecules, where the dots correspond to the higher weights. And then we have the measured molecule concentrations, and if we multiply these matrices, we get the simulated or the modeled responses of the neurons to the stimuli. And so the question that we want to ask are really about this tuning functions. And the question is, first of all, do the modeled responses fit the actual data that we measured in our electrophysiological experiments? And more importantly for this talk, are the repeated realizations of the modeled responses similar? So in other words, do we see a stereotypy in the responses that we get when we randomly assign weights to the neurons? In other words, when we randomly assign the tuning functions in the matrix T. So this is the focus of the motivation for this analysis. And the question then is, how do we evaluate whether we have similar or stereotypical responses? So this is the real data. This is an example of model data. And there are several measures that could be compared. So we could look at the population sparseness. We could look at the neuronal or the lifetime sparseness. We can look at the mean population response to each of the different stimuli. And we could also look at response categories. So for example, we can define a neuron that's female specific or male specific, or a neuron that's selective to a specific strain of mice. And we actually did all of these analysis, and we used them to compare the modeled and the real data. But I don't have time to talk about all of these. I want to talk about one measure, which is the distance between the population level representations. So by this, I mean just looking at different vectors, different columns here, that show the population response and to compare them to each other. And again, there are many ways, of course, to compare population level representations. We use various measures here. I want to show you the results that we obtained with the Euclidean and the standard Euclidean distances. So this would be a matrix of the pairwise distances between all these different stimuli. So of course, it's a symmetric matrix. We only care about one part below or above the diagonal. So we have these values for the real distances, for the real data, and then for the model data, and we can compare them. So essentially, we're correlating the 2 different measures of pairwise distances. And these are the results when we look at the similarity between the simulated data and the real data. And what this number is the correlation between the 2 response matrices, the 2 distance matrices that I showed you before. And we see the correlation, the mean correlation over repeated simulations, as a function of the number of sampled molecules. For example, here is 1 molecule sampled, and this is for 5 molecules sampled by each microcell, and so on. And the conclusion here is that, and it's a little bit surprising perhaps, that even with random sampling of molecules, we get some aspects of the data that are really fit well by the model. So this is one observation, and there are other measures that also reproduce well. But we know that this is the tuning functions are not completely random, and we know that, and there are also deviations from the real data. And the main question is whether the distances that we obtain upon repeated realizations, are they similar to each other? So this is really the question of stereotypy. So in this case, it's actually a very small simulation, just because I didn't think there was a need to do more. So I have 10 repeated simulations per condition, where I repeatedly sample these tuning functions. And so this means that we have 10 by 10. So we have 45 unique pairwise comparisons. And the question is whether they are similar to each other. Okay, when we all these 45 pairwise comparisons over the repeated realizations. And this is what you see here. So this is the mean value of the correlation between the distances across the repeated realizations, again, as a function of the number of molecules that each mitral cell samples. And we also see the spread of the individual points shown here in red. And so what you should also note here is that the axes don't start at 0, but they start at 0.8. So the correlations are very, very high. And one thing that we see that the mean increases and also the spread decreases as we increase the number of sampled molecules. And that's a good thing if we want to achieve stereotyping. Now, these results are with 237 neurons, and this is a number that we have in our neuronal data set. But we know that there are more neurons in the AOB. There are on the order of 6,000, maybe 7,000 projection neurons in the accessory olfactory bulb. And the question is, what happens when we use more neurons for this simulation? Do the simulations become more stereotypic? And the answer is definitely yes. So again, keeping the same axis, we see that the correlations are increased and the spread is much smaller, so the correlations are higher. And this is the key point really that I want to make here. So that with a sufficiently large population, and there are other measures of the distance between the model data that I did not show you here, everything becomes more stereotypic. And this is even under completely random sampling of the molecules and the glomeruli. So let me summarize what I try to show you here. First, that random sampling of molecules can replicate key features of the neuronal response. Second, that the random sampling yields the stereotyped representations across realizations. And I remind you that different realizations are like different individuals in this context. That is, different mice, for example, different wiring patterns in the AOB, which are supposedly random. And I showed you also that stereotypy improves with a number of neurons, and for some of the metrics, like those shown here, also with a number of sampled molecules per glomerulus. That's molecules per mitral cell. And after doing all these analysis and looking at this again and again, I asked myself whether this is surprising or trivial. And sometimes you begin by thinking it's surprising, and then you think it's actually all very trivial. And I think that if there really are enough unique cues associated with each of the stimuli that mice emit, and these are complex stimuli, right, urine stimuli, and if there are enough receptors to pick these unique cues up, and if there are enough mitral cells to sample the receptors and their combinations, then it's not so surprising. But maybe it is surprising that all these conditions are fulfilled. And now the last question is, what is all this good for? What is this? In what sense does this is this design good? I mean, wouldn't it be simpler just to have receptors for stereotyped components, for example, in predator urine, or in a different conspecific urine, parenting cues, reproductive cues, and just have labeled lines and respond to those? So yes, this is a good and simple solution, and it works. It would work for some signals, but not for all of the signals. And here we have to make a distinction between pheromones and signature mixes. So pheromones are predictable cues that elicit stereotype responses, and their detection and processing is probably simpler. But in contrast, we have signature mixes, which are unique combinations that are useful for discriminating among individuals. And we know, I have no time to show you, but we know that the vomeronasal system is really involved in various aspects of individual recognition. And you cannot predict these mixes in advance, or maybe like antigens, in the sense that you don't know what you will encounter in your lifetime. And also their significance is initially not known. And what the data shown here, or what I showed you here, indicates that the wiring of the AOB is suitable for discriminating these signature mixes and for maintaining the stereotype measures of similarity. And this also would allow mice to discriminate among individuals, and also maybe to assess the kinship or the relatedness of different individuals to each other. The very last thing that I want to say, of course, is that I've shown you a very small subset of the data analysis. In analysis, the real picture is more complex, and hopefully one day you will be able to read about this. And the last thing is just to acknowledge the people who are involved in this work. So most important is Rohini Bansal, who collected all the neuronal data for this experiment, and Max Speer and Max Nagel are collaborators in Aachen, who are part of this project, recording also from the vomeronasal system. And all the chemical data comes from Pavel and Romana at Charles University in Prague, who did the analysis and collection. And thanks to our funders, and thank you for bearing with me. I'm done.
Thank you very much, Iran. So Thomas has 2 questions, and I'll quickly ask him to come on stage. Just a second.
All right. Hello there.
Hello.
Yeah. So my question was about the assumptions of your model. You don't seem to assume that there was only a response to several orders at the receptor level. Is that correct? And is that what happens in the AOB?
No, it's not what happens at the AOB. I did a very gross simplification here in the sense that I equated the molecules with the glomeruli, and of course, we know that a given receptor can respond to multiple glomeruli. So in that sense, it's very much of a toy model. I don't think there's some evidence indicating that the receptors are very sensitive, but then there are other indications that receptors are broader than that. So I agree completely that this is something not taken into account by my model. But I have the feeling that the results, this is just a feeling that the results will not change much if we allow each glomerulus to respond to multiple molecules. In a way, it will maybe just effectively increase the number of molecules sampled by each microcell. And I should also add that my modeling here was completely simplistic. I mean, the linear summation is one option. It could be some kind of you may need some. There's no thresholding, for example, in my responses at all. And the tuning functions, we know them not to be entirely random. So this is the first step with many approximations and simplifications. But yeah.
Okay, thank you. Can I squeeze in the second one? Yeah. I also was wondering, you showed us results about distances, and they were stereotypic. How does that translate into stereotypic responses, or does it not?
So what do you mean by stereotypic response?
I mean, I would be surprised if the actual responses were stereotypic in some sense. I mean, you showed the distances between them are
right. So you're asking a stereotypic response would probably be that certain stimulus elicits maybe activity in the upper region of the AOB and a lower activity in the AOB. This I don't know, but I think this is a similar question that people ask about the piriform cortex. So it seems like the responses to a given stimuli are random across different individuals and maybe even across the different hemispheres, but you can maintain the distance relationships. This is the point that I want to make here. Our data there is some evidence for stereotypy in the responses. We know that there are subdivisions of the AOB. It's not something I can say about from this data, but I think it's for the analysis of different individuals, I don't think that stereotypic responses are important. I think that stereotypic distances are what is important.
The question is only how you get them innate responses from that.
Right. So you need something that's grounded you need some grounding. So for example, so that you know, so that you run away from a predator, or you know that this is. So I agree with you completely. It cannot be completely free of any grounding, and there's some. I don't have a good answer. I can only agree that this is a good question. Maybe one idea would be that maybe these more complex representations can be associated with simpler markers or tags saying this is a conspecific or this is a female or something like this. But it's a very good question.
Okay, thank you very much.
You.
Okay, yeah, thank you very much. So that's the end of this session, and we'll have a short break of 15 to 20 minutes, and then see you all again. Thank you. And you can also, you can go to Neurostars to look at more questions and answers for the talks. Yeah, thank you.
I think, hello everybody. I think we had a false start, so I'm going to reintroduce the speaker. My apologies. Are we live now? Anchor, can you just give me a wave? Okay, thank you. So my name is Mark Felus. I am joining you from the University of Arizona in Tucson. And it's my pleasure to moderate this session, which is on hippocampus. We have two great speakers today. I am joined as co-moderators by Gonzalo Cunio in Norway and Tom Burns in Japan. They will step in if something happens to my stream. In any case, we're a little bit late because of this false start, so I'm going to leave it to Andrea, who is from the Computer Science Department at Instituto Cajal in Madrid, to start her presentation on evolutionary algorithms to explore single-cell heterogeneity and microcircuits operation in the hippocampus. Andrea, up to you.
Thank you very much. So, hi everybody, again. I am Andrea Navas Olive. I am a PhD student at Laboratorio de Circuitos Neuronales in Madrid, Spain. Yeah. First of all, I want to thank the Organization for Computational Neuroscience for choosing my work for this feature talk. And I'm going to talk about how did we use evolutionary algorithms, well, a kind of evolutionary algorithms called genetic algorithms, to explore single-cell heterogeneity in hippocampus and how this heterogeneity is reflected at the microcircuit level. So let's start with this picture. Maybe it's not the best picture to start a talk, but why? Because it's too complex. And again, why? Because it has too many different elements in it. Well, and that's precisely why I want to start here. The hippocampus was thought to be a structure where the information flowed linearly from the entorhinal cortex to the dentate gyrus up to CA1. But this rapidly evolved into the picture you are now seeing. Not only shortcuts like these ones, or recurrent connections like these ones, were discovered. Also, cell type specific connectivity and functionality from many diverse cells. CA1 is the perfect example. It was not so many years ago when it was first shown that firing dynamics of pyramidal cells were different between the deep and the superficial layer. So the pyramidal cells from CA1 were divided into deep and superficial cells, both with different functionality, connectivity, and intrinsic properties. Here we have a genetic analysis of these two neuron types. We can see that gene expression in deep here and in superficial cells are different, being these genes responsible for both intrinsic and synaptic properties. Here we could ask ourselves, is this the mechanism underlying self-specific functionality? However, great models such as Besides, which is a realistic full-scale model of the CA1 microcircuit, meaning it has the same number of neurons as in a rat CA1, lack this heterogeneity. All modeled neurons from each population have the same intrinsic and synaptic properties. And this also happens in artificial neural networks, which most of them rely on identical units. But this is not all. During one of the most important hippocampal rhythms, the theta rhythm, a slow rhythm from 4 to 12 Hz, that is responsible for information processing during navigation, both inhibitory and excitatory inputs are distributed along all phases. In this picture, you can see the outside ring is representing excitation. And you can see how excitation covers almost all theta phases, from the peak to the trough and going on. And the middle ring is representing the inhibition, the interneurons, and they are across all the theta phases. Just a comment for later, all these populations, both the excitatory and inhibitory, are theta modulated. That means that its firing, it's more or less always in the same theta phase. For example, in here, we can see the entorhinal cortex layer 3 it almost fire its preferred phase, you can see that way, is around the theta peak. Okay, so in here we have a very high dimensional input from many different populations. And how is the neuron response? Given also all the heterogeneity we were saying before, not only for the pyramidal neurons, also all the different interneurons and also the excitatory inputs. How is the neuron response? It's bimodal. It's bimodal. So we saw that there are two main preferred theta phases, and we used a single cell recording and labeling in vivo in mice and rats. And what we saw is that the superficial neurons tend to fire in the trough of the stratum pyramidale theta. Here, you can see that all these lines are the firing along the different theta phases of each one of these, each one single cell, one neuron. And superficial cells tend to fire in the trough, while deep cells tend to fire in the peak. And we can see here the same. There are some neurons firing at the trough and some neurons firing at the peak. Here you can see the histogram of the preferred theta phases that are described with these white lines, and they are bimodal. A statistical analysis revealed that, they were separated also by cell-specific, cell-type specificity. So in the peak and in the trough, in the peak there were deep cells, and in the trough there were superficial cells. Okay, so we have here like a kind of cell-type specific dimensionality reduction performed by neurons, because they get a multidimensional excitatory and inhibitory input, and somehow they have a very low dimensional output. We think that heterogeneity at a cellular and microcircuit level play a key role understanding how the hippocampus operates, and that's why we decided to build a realistic model of the heterogeneous properties. We took a reconstructed neuron from Neuromorpho and performed a genetic algorithm looking for this heterogeneity. And the genetic algorithms goes as follows. We had several number of individuals, 45, and each individual is defined by a set of computational genes. In this example, gene 1 is represented the A potassium ion channel density and has one value, and the last gene is representing the axial resistance with another value. So we have a bunch of individuals with random genes starting in generation 1. We evaluated each individual with a given target function that returned a certain score. 30% of individuals with highest score remain for next generation. A random 20% of the remaining individuals was also retained. And the rest of them were completely new, like resetted with completely random genes. There's also a mutation rate. So 40% of random individuals suffered a mutation of one of their genes. That is, these genes were changed. And we keep this process on going from generation to generation up to generation 100. We ended up here with 45 individuals with very different sets of genes with high scores fitting their target function. Going into specifics, we took this genetic algorithm to constrain the intrinsic parameters that were ion channel densities and axial resistance also. The target function was an input-output curve that measures the spiking frequency given a certain current amplitude of current injection. So in green, you can see the target with certain variability. And these dots, connected dots, are the input-output curves of different individuals, of different individuals in this morphology. And the black ones are the ones that fitted the target function, the input-output target function, and the gray ones, the one that didn't, that has a low score. We also did the same thing. They applied the genetic algorithm to take the synaptic, to constrain the synaptic parameters, which were the maximal synaptic conductances. And for this, we took the result from my bibliography from this paper, in which CA3 was there was an excitation, a current injection in CA3 that activated some populations on CA1. And here we can see, again, like an input-output curve, but synaptic input-output curve. Sorry. The line green is the response for superficial neurons, and the red one for deep neurons. So we took this as our target function, which is this here in blue. And again, we can see how the different individuals were or inside the target function, here those in black, or outside the target function. These will probably be changed after some generations. Okay, so with here we have like a great pool of sets of intrinsic and synaptic parameters upon one morphology. We have a lot of diversity, but just in one morphology. We also want diversity in morphologies, as they are the bodies that perform the integration, the input integration. We did not perform this procedure, this same genetic algorithm procedure for another morphologies. We took another 3 morphologies because we wanted shared properties between all the morphologies to be able to compare them. So instead of that, we did the following. We took 20 random intrinsic sets from the fitted in the first the fitted pool from the first morphology, and we propagated them into the other 3 morphologies and evaluate them with the same intrinsic target function. If an individual didn't fit the target in all morphologies, then it was discarded, and we took another one from the pool. This was a validation process, an iterative one, until we end up with 20 individuals fitting the intrinsic input-output curve for all morphologies. We performed this same process with synaptic factors, although here it was very difficult to accomplish that all synaptic sets fitted the morphology. Fitted in all the morphologies the synaptic input-output curve. Sorry, so we were a bit more flexible. But now let's go to the fun part. How do these synthetic cells behave in an oscillatory regime? In order to simulate theta oscillations, we distributed the synapse connections of both the excitatory and inhibitory populations in a realistic way. So not only synapses were distributed along the morphology according to bibliography, we also the time distribution was fitting the experiments. Before I told you that most of the populations were theta modulated, meaning that they had like a preferred phase. And here we can see, for example, the PB interneuron mostly fires in the descending part of the theta rhythm, as CA3, for example, CA3 also fires mostly in the descending part, different from CA2 that mostly fires, for example, in the trough. Here we have all the inputs, the excitatory and the inhibitory inputs, and here we have the output from one of our synthetic cells. In black, you can see the membrane potential of the soma, and the lighter the gray of these traces is further in the apical dendrite. We can see, obviously, these spikes, but we can see also some other interesting properties of these traces. For example, backpropagation. So here we have a spike, and the spike propagates into the apical dendrite, like in here. We also have dendritic spikes, as in here, and we also had dendritic spikes that provoked a spike, firing in the cell. Okay, so but here we have one of our synthetic cells. How about we see more? If instead of looking at the traces along time, we take the firing histogram, the spiking histogram, we can see in here different spiking histograms for the same individual. That means it has the same intrinsic properties, but across different morphologies, our 4 morphologies. We can see in the bottom the firing histogram, and in the upper part the membrane potential, in the soma black, then in the middle, and the lighter is the distal apical dendrite. And what we see is that even the intrinsic properties are the same, integration is not, because the output is different. So here we can see that this morphology performs a bimodality output, while this one and also this one mostly fires around the latter part of the descending slope or the trough. And this one, just the opposite, it fires mostly in the peak. And we can see also that the traces reflect this same spiking. Here I've just shown one individual. Let's see more. The individual from the slide before is this second row. Other individuals are in other rows. That means that all these rows, so this row has the same intrinsic properties, different from the intrinsic properties of this row that are different from this one. Each row is one individual, and in different morphologies. Each of these columns is a different morphology. And what we see here, more or less the same as before, there's variability not only across morphologies, also within one morphology across different individuals. So here, for example, we can see this morphology, it depends on the individual, it fires at the peak or at the trough, or almost doesn't fire at all. We can see the same in the individual level. We can see that these intrinsic properties make these two synthetic cells almost silent, while these ones in this morphology it almost fires almost always at the peak, and here in the descending part. And it's the opposite in here, which these neurons fire less, and in here we have one in the trough, in the peak, and one in the trough. Another way to see this more compacted is taking the preferred phase that is computed as the maximum of each of these histograms across the different morphologies, and see how is a histogram of the preferred phases for each morphology. And here are the results. We can see that most of the preferred phases are or in the trough, or in the peak, trough, and trough. So here we have an example of a non-fitted phenomenon, the bimodality we saw in the experimental data coming along. Maybe this is not enough to validate this model. We can go one step further and test the model with a prediction. We know from experiments that the CCK and PB basket cell interneurons connect differently with superficial and deep cells. In particular, CCK basket cells innervate more superficial and less deep, and PB basket cells innervates more deep and less superficial. And we also know that the CCK basket cells fires mostly in the peak, while PB basket cells mostly fire in the descending part. We can perform a computational experiment trying to seeking like a deep-like synthetic cell, lowering the CCK. We could do also increase in PV, but if we increase inhibition, then the model won't fire, or we will need a lot of more time to achieve the same number of spikes. So we decided to lower the CCK to do deep-like cells and lower the PV to do soup-like, superficial-like cells. Here we have the mean membrane potential. And well, I haven't said that these are perisomatic inhibition. So here we can see that the membrane potential in the apical dendrite is almost the same for both experiments, lowering CCK or lowering PV. But there's we can see that here at the somatic level there's a difference. So when we lower CCK, the neuron fires where the CCK was supposed to be, in the peak. And when we lower PV, then the neuron tends to fire where the PV was supposed to be, in the descending part, making these two neurons like deep because it fires mostly in the peak, or soup-like because it fires mostly in the trough. We can see this in a more nicely way in spiking histograms. In black, we can see like the basal state where there's no lowering. And in blue, we see the lowering CCK that fires mostly, as we said, in the peak, and lowering the PV basket cell that then it fires mostly in the descending part, or even at the trough. In the bottom, it's not related with this PV CCK, if you want to see it that way, but in fact, yes, it is related. Here we removed all inhibition, not only CCK and PV, but all inhibition. There's just excitation, and you see that there's like a more or less intrinsic bimodality without inhibition, and what maybe CCK and PV is doing upon this no inhibition state is choosing between one of these two windows of opportunity. So when CCK is present, then this window of opportunity is chosen, and vice versa. Before, I told you about the full-scale Bethayre model, that it's very cool, not only because it's full-scale, but also because the theta rhythm is generated spontaneously with tonic excitation, without rhythmic excitation. We wanted to see if the CCK and PV lowering in this model also gave rise into deep-like and soup-like cells. For that, we used the Neuroscience Gateway, that is a supercomputing center that is free for neuroscience research and very easy to use. I really liked it and recommend it. And here we can see the results. We performed the experiment lowering CCK and PV. But we couldn't get these superficial and deep-like cells. We were quite surprised and disappointed also. But later we realized that the local field potential, the clock by which we measure the theta phase, is computed from pyramidal cell spiking. So by definition, pyramidal cells are going to be firing always at the trough. So we couldn't use this tonic excitation input. We had to use rhythmic excitatory inputs to have an external clock. And doing that, we had what we were expecting: superficial-like cells firing mostly in depth. This is a raster plot, sorry. So each of these rows is one different neuron, and we have here, I don't know, thousands of neurons. While I am using just one pyramidal cell, and what pyramidal cell at a time, sorry. So here we see that the superficial-like cells fire mostly at the trough, and the deep-like cells fire mostly like in the peak. But there is no testing without an experiment. So the experiment we were going to perform was turning in the same line as we were talking, turning down the PV interneurons in the model. Here we can see each of these rows is the spiking histogram of one synthetic cell with different all this heterogeneity we have been talking before, and the preferred phases are the ones plotted with this black line, and we can see the distribution, the preferred theta phase distribution in here in the bottom panel, and as we could expect, is in the descending part of the theta rhythm. The experiment was the non-computational experiment was this same thing. We silenced the PV interneurons, the interneurons expressing PV in PV creme mice, and the results were in both juxtacellular condition with this setup and a multi-site setup. Again, each of these rows is a different neuron. The spiking histogram along the theta phase of different neurons. And we can see in the bottom panel the preferred theta phase histogram. So if we compare both distributions, they are statistically the same. So with this, we can now be confident that our model is working. It can predict experimental outputs, and it also has achieved non-fitted phenomenon as the bimodality. With all this, we can go back to the basal condition and now study deeper how are the different factors contributing to the output. In here we have, in this panel, we are going to be first looking at this panel. We have the different individuals, the preferred phases of the synthetic cells sorted by different individuals. Each of these 4 dots are the 4 morphologies, and they are sorted by the mean preferred theta phase. And what we see here is the expression level of the intrinsic parameters defining each of these individuals. So, for example, this individual has and if it's green, then the expression level is high. It means the value of these genes are high, and it is yellow. It means that the value of these factors is low. In this individual, for example, is defined by a very high potassium channels, CKDR A potassium channels, also high leakage current, and also a high sodium channel, but it has a lower calcium channel and a low M potassium ion density channel. What we saw after comparing the different preferred phases with the different factors is that there is a significant correlation between the phase preference and the expression level of the intrinsic factors. We can see, for example, the C potassium ion density channel, the higher it is, the higher is its expression, the more nearer to the peak the preferred phase is. And while it goes down, it lowers, then the preferred phase tends to go to the trough. We can see that this is shared with other intrinsic parameters, but there's quite the opposite. For example, the axial resistance. The lower the axial resistance is, the nearer to the peak is the output solution of the neuron. And while it's high, then the neuron tends to integrate somehow the inputs, doing the synthetic cell fire in the trough. And well, just a comment, here you can see the correlation coefficients, and here the p-values. So this is very like very statistic. It has a very high weight in statistical in a statistical sense. We have seen here the intrinsic parameters. Let's go to the morphological and cymatic parameters. Instead of being now sorted by individuals, it's sorted by morphologies, and the dots represent the different individuals. And here we have the level of expression of different factors, of the excitatory factors, inhibitory and morphological, that is the number of branches of each of these neurons. And here we see that there's a statistical correlation in the number of branches and also in the inhibition. Here I think it's pretty clear the number of branches, because the less number of branches it has, the morphology, near to the peak is going to fire. So again, here this picture is showing that the different intrinsic, also cymatic and morphological factors are determining the phase preference of the neuron and somehow contributing to this lower dimension output. We can go a step further and see the same kind of analysis, but with taking into account also not only the basal state, but the experiments we did, for example, with the CCK and the PB. And the way of reading this is which is the preferred theta phase when you downregulate this factor, and how is the theta the preferred theta phase when you upregulate this factor. Here we can see the results of the CCK and PB experiments. If we lower the CCK, then the neurons will start to fire in the peak. And if we lower the PB, then the neuron tends to fire in the descending part. We can see that there's, I haven't said this, but these are the results that are significantly statistically significant. We can see that there are more interneurons that contributes to the phase output, like the OLM, also some excitatory inputs, and also what I like, for example, here the one potassium channel. But what I really liked is this sodium channel, because it's super intuitive. How is this working? If we have a high sodium ion channel density, then first of all, if we remind, the excitation starts coming into the theta phase in the peak with entorhinal cortex layer 3. Then it comes CA3 in the descending part, and it ends with CA2 and entorhinal cortex layer 2 in the trough. So if we have a high value of a high sodium conductance, then as soon as any excitatory input comes, the neuron will start firing, and that's why it fires at the peak. And but if it's very low, then it's going to be a silent neuron, and it needs all the excitation across the different phases to finally fire at the trough. Here we can see again the results from the previous slide, that also the morphological properties are somehow modeling this low output, the low dimensional output. So with all this is showing that the mechanisms underlying the neural activity are multidimensional. There is a natural dimensionality reduction performed by neurons and fostered by the nonlinear interaction of multiple factors, including the intrinsic, like the sodium and the potassium, and morphological, such as the number of dendrites. And I like to see this as a reminder of the importance of studying the circuit with all its components, and that we have to make use of these super cool cutting-edge technologies that are coming to, instead of studying the circuit its properties in isolation, try to study them like a whole picture. I also wanted to say that this model is now on GitHub. My GitHub is my name, basically. And then the model is this one, the Laboratorio de Circuitos Neuronales HIPPO model. It runs in Python. I use the Neuron software, but it's code is Python. And there is a nice documentation also in the GitHub page. So finally, I wanted to thank, well, everybody in the lab, especially my supervisor, Lisette Menendez de la Prida, and all my colleagues performing the experiments, and of course, all you for your attention.
Thank you, Andrea. That was an impressive talk, and thank you for sharing all your software on GitHub. We appreciate it. It's really useful for the community. So we have tons of questions. So I am going to try and invite a few people on stage, if I technically can do that. So the first one will be Alexandra. So let me see if I can do that without crashing everything.
I see now the...
Yeah, everybody's...
yeah, no, it's nice, I mean.
All right, so I'm inviting Alexandra. Let's see. Alexandra, I should see your message saying if you want to come online.
Oh.
Right.
Oh, yes, here I am. Can you guys hear me?
You are online, yeah.
Yes.
So,
first of all, congratulations. Very impressive work. I've looked at your paper. This is really amazing, what you've done. I had a few questions just to clarify certain aspects. So you mentioned that, I'll start from the first one. Oh, I just changed the order because of the votes. So yeah, so the first one, I was wondering whether you looked whether any of your cells are not biologically realistic as they were derived from the evolutionary algorithm. So did you just use the algorithm to create a bunch of cells and then use them all in the model, or did you test whether any of them were biologically unrealistic to include in the model?
Well, for performing the genetic algorithm, we also had like certain restrictions, so the values won't become like super unrealistic. So
okay.
we didn't, yeah. Apart from that, we didn't like see.
I see.
I don't know. Yeah.
Yeah. No, I understand. And what does lower mean when you said you said lowering the PV cells, lowering the CCK cells? I don't know what that means in terms of the code, like removing the inputs from those cells to other cells. Yeah, exactly that slide.
Yeah. So yeah, in the one of our works, we saw that the number of connections between CCK and PV was like, for example, with superficials. If you say that there's 100% on the superficial, then it's going to be like a 30% on the deep, and vice versa. So there's this 30 and 100%.
So it's a connectivity.
Yeah.
Yeah, it's a connectivity.
And we also performed an experiment seeing if the connectivity and the conductivity, like the number of synapses and the conductance, was equivalent, and it was. So we could have done it like either way.
I see. And when you changed, so those parameters, when you changed them, you knew from experiment that it was okay to sort of lower those connections by 30% or to increase them? Like when you said like from 100 you went to 70, did you know that you could make those manipulations from experimental observations, or you just
No, so this like 30 and 100 were from experimental observations.
Okay.
Yeah.
All right.
From the
Okay.
Sorry.
Okay, great. And the first question that I asked, I didn't understand how the genes corresponded to different intrinsic properties in the pyramidal cell. So when you first designed your evolutionary algorithm, you say you changed various genes, but I don't understand how that maps on the model. Because you used Bézer's model, like that doesn't have any genes, of course. It's the intrinsic property that,
Yeah, so for example, you have this morphology, and then let's see, the axial resistance. It has to be something. It has to have a value. So that value was the value from the genetic algorithm. And also you have to have a conductivity, for example, a sodium or a potassium conductivity to so the different channels can work.
Right.
So these numbers were from the genetic algorithm. The numbers were like were on the, how I say, can I say this? Yeah, so they went into the morphology in terms of the like Hodgkin-Huxley equation parameters.
Right. Now I understand that. I thought you said genes though, and I thought somehow real genes were involved in this process.
No.
Okay,
It's because, yeah, so here we have that the genetic algorithm, it's called like that because it comes from the like evolutionary Darwin.
So genes in quotations.
Yeah, so so it's like computational genes that I thought I did this, but of course you won't see me doing this now, no? Yeah.
And there is just another question under my comment by, that says, moreover, what is the distribution across the dendritic trees and soma? By Maria Psaru under my comment.
But sorry, can you repeat?
Yeah, so Maria Psaru under my comment, she asks, what is the distribution across the dendritic trees and soma of those?
The distribution of
the physical properties, I suppose, the ion channels.
Okay, so I had like I cannot like remember perfectly, but from bibliography we had like at the soma there's this quantity, and then it reduces to the 50% in the apical dendrite, and then it goes down to a 20% in the distal apical dendrite. So we like multiplied this all because if not, you have like thousands of parameters to fit with the genetic algorithm, and that cannot. I mean, that doesn't work. So we did we maintained like the distribution along the morphology. We maintained that and multiplied it by the whatever was in the genetic algorithm.
Let's see. Okay, thank you very much.
Thank you. So we have a few more questions. Let's see. Laura had a question. I'm not sure if she wants to come on stage. I asked, but I didn't get a response. So let me read the question. So the choice of the objective function to select and generate the individuals at each step is critical in genetic algorithms, especially when there are several target output to satisfy. What is the mathematical expression of your objective function, and how much fine-tuning did you have to do to get the results? So this is about the objective function and how did you choose it and what is it?
Well, I suppose objective function means like what I've been calling target function. I think
so. It's a cost function, right? So how do you decide?
Yeah. So if I recall this properly, I think we used like minimum square error. So at first we wanted just to fit like a curve without any error, and that won't work because it's super difficult. So we went to we also integrate into the target the target function. Let me see if I can. This we like increase this width because also in experiments there's variability. And with this, we didn't have to do any much working because after many generations, the neurons end up choosing this, like fitting this target function. And I think the optimization was we used minimal square error.
Great, thank you. And then one last question. This one is from Dieter Jäger. Is there any specific function for NMDA versus AMPA current in the excitatory inputs? And same thing for GABA A, GABA B.
Yeah, so that one thing that we wanted to try, but we didn't get to that point. I think we used only GABA A, and also we didn't get to add how do you call it?
NMDA.
Yeah, sorry.
Only fast synaptic transmission. So you had very
Yeah.
fast synaptic transmission in this model. Okay, got it.
Yeah.
Great, thank you very much, Andrea. Great job. And I will drop you from the screen and invite our next speaker, Nathan Schultheis from Florida International University, the psychology department. So let's see if I can find Nathan. All right, and we go. So we're running a little bit late. I hope that's okay, 5-10 minutes. Nathan, take the time that you normally have. Don't worry about the absolute time. The next session is a poster session on the hour, so we have a little bit of time. Nathan?
Let's see, am I here?
There you go. I can see you now.
Great.
Great, welcome.
Thank you, thank you.
Try and share your screen so we can get started.
Yeah. Let me, okay.
So we don't see
Let's see, is this,
so it should be.
I don't know why it's, doesn't seem to want to let me do that. So if I just,
All right, so you disappeared. I'm going to, again, screen invite you. All right, we can see you.
okay. If I go to share screen again, am I coming, is my audio coming through?
Your audio is fine. We can see your video stream. We cannot see
Okay.
your slides.
So, sorry.
Okay, it worked when you practiced it.
Yeah, it sure did. Okay.
Whatever you did then.
Yep, so now it started responding. So do you see my mouse on the shared screen right now?
I do not see your shared screen.
Oh, you don't? Jeez.
Nope. I can see you.
Okay. When I click on share screen through the app or through the browser, it doesn't respond.
All right, so we
What
have
was the other?
some admins online. If they can help out, that'd be great. I don't know what to do here since I have you. I have share screen for me, but that's not what we want.
So do you see, you don't see anything on my computer at all, right?
All right, so somebody on the chat window says you may want to reset your browser.
Okay, so shall I just close that and then start over again?
Yes, you go ahead, and I will.
Okay, thanks.
Maintain the link. All right, my apologies to everybody. It's just a little glitch. So Nathan is restarting his browser. Yeah, reloading might not be the best. But it should be just a matter of a minute.
So I'm back.
Then we have you back. We'll try to reshare your screen again, maybe.
Yeah. Why? Now it's showing me. Oh, this is unfortunate. Now I'm unable to get certain Crowdcast windows off of the screen here. There we go. Okay, let's try this. I do have a little red. Ah, here we go. Okay. Let's see if this works.
All right. So now,
Okay, see, does that work?
No, I can't see anything.
It says it's thinking now, where it used to say just share screen.
Okay, you're on. Whatever you do.
Great. Okay, sorry about all this.
Okay. You want to go into full screen mode, maybe?
Yeah, for sure.
And I'll let you just take it away.
All right. If it will actually respond now, that would be great. Okay, one quick question. Do you see my mouse on the screen?
Okay, how about you jiggle your mouse a little bit, see if I can see it.
I'm moving it back and forth. What about here? No?
I do not see the mouse.
Okay, well, I'll just try to use verbal cues as best I can. Okay.
And actually, we're not seeing your full presentation. This is still the...
Is this working?
So this is not full screen. So you're probably sharing the wrong screen.
Yeah. So swap presenter and slide. Seems like it should work. How about now?
All right. It's still
It's just showing the presenter.
This is not a presenter mode. Now this is the, we can see all the slides on the left hand side. And
oh, really? I can't even see that. That's interesting. Okay.
You should share your entire screen, somebody said. That may help.
Yeah, let me see if it will ask me what share. Okay. Screen. Let's go with screen 1. Okay, should be thinking now.
Okay, so this is the same as before.
I do not know why this worked the other day. And
right, so it's not too bad if you just maximize your slides quite a bit. Like take out maybe the right-hand side if you can. I'm not sure if you can move the short of, yeah, right. It's not ideal,
right.
But if you can do it, then maybe we should do it.
Yeah, it's and this is still showing you that same piece, right?
No, this is better.
Is it full screen now?
No, this is now we can see the slides and then your next animation on the side.
I see. Okay, so then let me try this and see if this makes everything happy.
Ah, you got it. This is
okay, great. All right, so you can see me, you can see the talk, correct?
Correct.
Wonderful. Okay, so good morning or good afternoon, depending upon where in the world you are. This is actually the first time I've given this talk, and so I thank the organizers of CNS for giving me the opportunity. First of all, this work is done in collaboration between Tom Galarte's lab here at Florida International University, FIU, And this work was actually done in Tim Allen's lab. I work for Tim. And the Galarte lab is primarily focused on neurotoxicology, whereas Tim Allen's lab is focused on neurobiology of cognition using a range of electrophysiology and circuit manipulation techniques. So there we go. Seems to be advancing at a strange rate. So, so my title is a little bit of a mouthful, but the paper on this work, today's work, is going to be coming out in Behavioral Neuroscience very soon. If you want to access to preprint, then maybe note down that number there. And with the Galarte lab, we also have a companion paper on the effects of lead neurotoxicity on some of the things I'm going to be talking about today. So this is a bit of a mouthful of a title. So first of all, let me break it down for you. I'm going to be focused on what we're calling, for our purposes, awake delta and the theta rhythm. And these are something that I like to refer to as modes of hippocampal network activity. And then I'm going to be looking at these modes, or talking about these modes during just basic locomotor behavior in the rat. So by way of introduction, this is a depiction of the theta rhythm. If you could just let me know if you can actually see my mouse currently. Is this showing up? Well, I'm not hearing anything. So, these traces are recorded from a depth probe
we can see it. Don't worry. We're good.
Sorry?
We can see.
Okay. Okay, wonderful. Thank you. This is a depth probe used to record the local field potential at various depths throughout CA1. Up here at the top, the pyramidal cell layer is about here, down into the stratum lacunosum moleculare and into the dentate gyrus below. So you can see that there's a profile of the theta rhythm, which is pretty well known. I would expect that most of us are somewhat familiar with it. And for our purposes right now, we're defining the theta rhythm as 6 to 10 hertz. Delta is a bit of a more complicated thing to discuss because the field has a lot of different perspectives on it. One of which is, again, from the Bizaki lab, a 4 hertz oscillation that's within what we would call delta, which is the 1 to 4 hertz frequency band. You can see that a 4 hertz oscillation emerges in PFC and the prefrontal cortex and the ventral tegmental area during a working memory task. There's also a very different definition of delta, which is during slow wave sleep. So polymorphic slow wave sleep looks like this. These are local field potential recordings. And in each one of these columns here, you can see depicted a delta wave, sort of a singular event. And the way this has been typically conceived is as a moment of cortical silence. Now, if you look at rasters from prefrontal cortex that are depicted below here, you'll notice that there's really no spiking in prefrontal cortex during these delta waves. And so it's been thought of maybe a reset period or for whatever reason, a moment of silence. However, very recently it's been shown, as you can see in this circled region right here, that sometimes spikes occur. So you can call those delta spikes, if you will. This is from Todorova and Zugaro just recently, a few months ago. And actually an additional figure from that paper shows 3 different delta waves that have delta spikes occurring during them. And what the authors show in a variety of ways is that hippocampal activity prior to these delta waves is predictive of which neurons are active during those delta waves. And this can be shown to be related to subsequent memory. So you have an awake version and a sleep version. There are differences in frequency band that I'm going to talk about. But first of all, let me give you a sense of what our setup was. We are recording local field potentials. Everything I'm going to talk about today is local field potential. So the way that we do that is we have a rat running around in what we call an open field environment while we take video of him from 2 cameras above. And in this case, the rat has no task, just completely free navigation, wandering around this environment. And you can see on the floor here during a roughly 40-minute session everywhere that the rat went. This is called the rat's trajectory, not surprisingly. And this would be a session that had really good coverage, meaning the rat went everywhere within that environment. This is an actual photograph of the environment. We dimly illuminate it during recording sessions so that it's red. Rats don't see red very well, and I'm not sure if you can make this out, but there are black curtains that go floor to ceiling on either side. The experimenter has essentially no interaction with the rat, and like I said, there's no task. So hanging here from the recording apparatus above is a head stage that contains green and blue LEDs that we use to track the head of the animal. This is what our tracking actually looks like. So you can see with the two cameras that we use, sort of, I call this a dark field and a light field camera. You can track the LEDs on the rat's head with this camera, and we track the rat's body with the second camera. And then we map these two cameras onto one another, so we have a momentary estimate of where the rat's body is and where the rat's head is at all times. That is relevant for a piece of data I'll show you at the very end if I have time. So our recordings are done with NeuroNexus probes. These are silicon probes that have 8 tetrodes on them, distributed across 4 shanks. So we have 2 different depths where we're recording from on each of 4 positions that are oriented medial lateral or along the proximal distal axis of CA1. A second recording setup that we use that is relevant to one additional figure I'll also try to show you before the end is these are handcrafted or custom made, I should say, in the lab. They're 3D printed recording bodies that have an electrode interface board in them, and then any conformation of electrodes that we'd like to record from. So in these recordings, we have 18 hippocampal electrodes and then 14 in medial prefrontal cortex. And that could be printed out in, like I said, any conformation. And once we are recording, after the experiment's over, we can do the histology, and you see these 8 red circles here, hopefully, that reflect the 8 recording sites from a silicon probe. And these are some dual recording, a brain that had dual recordings done, and you can see a few hippocampal sites for these stainless steel electrodes and a few prefrontal cortex sites. Okay, so this is what the data basically looks like, and I'm also going to use it as a bit of a summary slide. So this is the local field potential, just an example trace that we recorded. And local field potential, of course, reflects synchrony among membrane currents, which are largely synaptic currents, and therefore they reflect the local network activity as well as potentially long-range inputs. If we filter this raw LFP trace in different frequency bands, we can see the theta rhythm emerge, and we can see delta frequency activity. And we can plot this as a function of the movement speed of the animal. So local field potential. So in a nutshell, what I'm going to be talking about today is a relationship, or sort of dual relationships between theta and delta and the behavior of the animal. So what you can see in a nutshell part here is that theta is really prominent when the animal tends to be moving around, but in moments where the rat stops moving and is very still, you can see sort of this coincident, if not synchronous, emergence of delta at those moments. So this is more of a theta half of a trace, and this is more of a delta half of a trace. But the way that rats actually run around when they're not doing a task is this sort of, I call it creeping. They'll run a few steps, or maybe several steps if they have somewhere that they're trying to get to, but they'll pause very often. And during these pauses is when the delta pops up almost immediately. And that is an observation that at least hasn't been treated very strongly in the literature. So just to give you an additional...
Sorry to interrupt, but we can see your mouse now if you want to use it. Feel free.
Okay, right here? You see me going in a circle with the mouse?
Oh,
no?
You did a minute ago.
Just for a moment. Maybe it's a refresh thing, because I've actually been using the mouse to point, so hopefully I haven't lost...
Feel free to use it anyway if you like, but sometimes we do see it.
Okay, so hopefully, yeah, it's actually flashing for me now. So maybe this is just a memory issue or something like that. But in any case, I'll try to be a little bit more conscious of describing verbally instead of assuming that you see where I'm pointing. So the question becomes, if delta is in fact related to the moments in time where the rat is stationary, and theta is related to the moments in time when the rat is running, do these different hippocampal modes of activity, or just the synchrony profile, reflect different things cognitively? So is if you will, is the rat thinking about something different when he stops and pauses amidst the intermittent pattern of his running behaviors? So let me do another, in a nutshell, image here, the idea being to just show you the thing. If it's there, you believe me. If it's not there, you don't believe me. But if you look at the spectrogram, which shows the power and the different frequency bands across a recording session, this is about 20 minutes of a recording session, and I've overlaid the running speed of the animal here in white. What you can see is that when the running speed goes big, when it goes up, there tend to be really red dots here in the spectrogram in the theta band. So this is theta indicated at about 8 hertz. And when the rat stops moving, you see what I think of this looking like a rainstorm or something like that. You see delta raining down from theta at these moments. Now, there's a couple of caveats to looking at a spectrogram. One is that you have a time window across which the power spectrum is taken. And so this is not going to be on the time scale of fractions of a second, but rather 2 seconds in this case for each time window, which approaches how rapidly a rat can change his behavior, but is not necessarily perfect. So it's a good idea to do some of these analyses in multiple ways. So taking the longer time scale approach, we can see that both delta and theta appear to be related in some way to movement speed. So the power spectral density across the entire session shows activity in both of those frequency bands. You see this really large theta peak, and you see sort of a shoulder, or in some cases a distinct peak in the delta band. However, if you focus on smaller time windows than the entire session, for example, the red and the blue that I've highlighted below here, sorry, the green and the blue that I've highlighted below here, during the green window, the animal is running, and during the blue during the blue window, the animal is very stationary. And you can see that the frequency composition of the local field potential is very different, with there being more power in delta when the animal is stationary and more power in theta when the animal is running. Now, the delta trace here looks like it's quite distributed over a number of different frequency bands, and of course it is, and you can look at the window and see that within the spectrogram, the time frame of that moment. But it's a long time window. These windows are 36 seconds long, and so activity at different moments in time is not captured by the PSD from those longer time windows. So this is just another example. So you can see these rain showers coming down. This animal's running a lot more consistently, so you see a lot more theta throughout this session. And again, this is when an animal doesn't have a particular task. Some animals tend to be very exploratory, and others tend to be relatively sedentary. So, like I said, the PSD is not necessarily the only way that we want to look at these things. If we look at the raw LFP filtered in the different frequency bands, delta, theta, and fast gamma in this instance, we can take the peaks and troughs of every peak and trough of this signal throughout the session, throughout these filtered versions of the local field potential, and plot the amplitude, the peak to trough amplitude for every half cycle, right? So the left and right side of each oscillatory cycle. You can see that if we do that, these dots in delta, for example, are going up, and then they drop down towards the middle, and they go up again, and they drop down. So if your eye can follow that in the trace below, the filtered trace below, it obviously gives a nice readout of what the momentary amplitude of delta, theta, and fast gamma are. And if we take small time windows or large time windows, I believe this is 1 second time windows, and take the average amplitude within a sliding 1 second window as the animal is running around, we see that delta seems to have a negative relationship to running speed, which makes sense based on what we saw in the spectrogram, whereas theta and high gamma are positively correlated. Theta and high gamma have been known to be positively correlated with running speed for a long time. That information has been used in a number of models of spatial navigation, dead reckoning, if some of these terms are familiar to folks. But the spatial navigation literature uses theta for quite a number of different things. But in general, I would conceive of it as a coordinating influence within hippocampus and also between parahippocampal structures and prefrontal cortex. So it's a coordinating influence. What delta is doing here is not known currently. It's not been observed to be modulated by running speed, but this, maybe it is, maybe it isn't. However, in this case, when we analyze theta in the same way, we do see a similar relationship between running speed and delta. This is just a second example of the same thing, where animals run less often, as you can see from the trajectory plot, but faster when he does. And these are all male rats, so I say he. So what we can do by taking extractions from all of our recording sites, so we're using a 32-channel system, and we have 6 rats in this data. So we end up with whatever that multiplied is, number of channels. So we can take for each channel that we recorded from the correlation coefficient between running and between running speed and each of those rhythms, the amplitude of each of those rhythms. So across all of those 150 or however many recording channels that comes out to be, we see that the correlation coefficients are almost entirely negative for delta. So this is just a really strong way of saying that almost always this relationship between running speed and delta is true. We don't see a slow gamma effect. That's a very complicated subject right now in the field. But we see very positive relationships between running speed and theta and running speed and fast gamma. As I said, that's already known. If we do a 4 predictor regression where we predict running speed on each of those 4 rhythms, then, or I should say oscillatory frequency bands, rhythm can be somewhat of a loaded term, but we get beta values for each of those oscillatory bands. And again, the delta and actually slow gamma in this case show negative relationships to running speed, and theta and fast gamma show positive relationships in terms of the betas from that regression analysis. Now, this is just a caveat. What I want to say here is that this is by no means a complete story of what's going on with the different rhythms. We saw considerable variability between recording sites for the relationships between rhythms. So in this case, the black plots here at the bottom are just histograms of the correlation coefficients, similar to what I already showed you, but in this case, between the different rhythms. So theta to gamma on the left, delta to theta in the middle, and delta to gamma on the right. And you can see multiple peaks in these cases. So what that suggests is the possibility that the different rhythms or the different frequency bands have different relationships to one another under different circumstances. So if 3 different things are happening, you might expect to see 3 different peaks in the relationships between the different frequency bands. So I'm not going to go into this in greater detail just because it could go on forever. But in addition to the many different relationships that may exist between different frequency bands, we can already see that there's a lot of things going on in the spectrogram. So this is just another example of a spectrogram showing the rain showers, like I was telling you. But if you look with a fine detail, you see that in some cases theta is very high. In some cases there's what seems to be a combination of theta and delta. In other cases, purely delta. So I naively wanted to ask how many different things are going on in the spectrogram. So if you take 2-second windows and run and calculate the power spectral density from all 2-second windows throughout a recording session, I then applied a clustering algorithm to that to, in a naive way, allow it to tell me how many different things are going on. Now, in this case, I'm just using the linkage function, which is a built-in MATLAB function. But really, any clustering algorithm can be used with different caveats. I've tried several, but there's quite a bit more work that could be done on this. These are just 2 examples that show a whole bunch of modes, if you will, or instantaneous power spectral densities, I should say, that have a theta peak, and many of them also have a delta peak that's off to the left of these plots. And there's some variability within animals and some variability between animals, but they almost all show this general profile of a whole bunch of stuff happening in theta and a whole bunch of stuff happening in delta. If I then cluster those based on the dendrograms that are inset here, I get different modes, is what I would call them, spectral modes, that have different frequency compositions, basically different patterns of behavior. So for those 2 examples I just showed you, on the left here are 16 different putative spectral modes, you might say, and you see that they tend to fall either in the delta range, or the peaks of power tend to fall either in the delta range or in the theta range. I plotted those on top of each other here on the right. But now, from those different categorizations of PSDs, I'll say it again in a different way, from those different modes that were given to me simply naively by clustering the spectrogram, I get measures that I can make for the different frequency bands. And so here is one of the main points of the paper that I mentioned earlier, and it's that delta power and theta power across modes appear to be orthogonal, meaning essentially that there's a right angle. between the power in the two bands. So the way I would describe that is that all of the variants, if you will, all of the different values that delta power takes on occur at the lowest values of theta, whereas all of the larger inflated, accentuated values of theta occur when delta is very weak. So this would be an orthogonality between the two. So the other, an initial way that this kind of analysis is useful is because, or a second way I should say, is not only can you look at the average behavior of the power spectral density itself, but what a mode is all of the instances in time where a particular pattern or where a power spectral density that was categorized in a particular way occurred. So it's the incidence of all of those moments. And if I want the average power spectrum, I just take it across all of those instances. But I can also take other values as a at all of those times to compare those. So a couple of the last things I want to show you here, actually, yes, the very last things I want to show you here are if we take all of those moments and we plot intrahippocampal delta power, then we see this orthogonality that I was describing before, because when the animal runs fast, we tend to get theta and low delta, and when the animal's moving slowly, we see delta. And theta power correlates with movement speed across these modes. But more interestingly, perhaps, when we take the medial prefrontal cortex to hippocampus coherence, we see a monotonic relationship in between each of these rhythms and the theta delta power ratio, which is a psychiatric tool that's used in a number of cases. But so what this says is that delta coherence decreases, delta coherence between medial prefrontal cortex and hippocampus decreases as the theta delta ratio increases. So when you have, and the theta coherence increases with a larger theta delta power ratio. So what this suggests is that the interaction between hippocampus and prefrontal cortex, the interaction between the two follows the actual spectral content themselves. So when we see modes that have a lot of delta, the delta coherence tends to be higher, and when between the two structures, and when we have a lot of theta, the theta coherence tends to be higher. So the take-home messages are simply that delta may be functionally important for episodic memory. In this case, I didn't talk about episodic memory at all, but if you imagine one of these intermittent trajectories of locomotion, that is essentially conceivable as different events that are strung together. And so if during those different events strung together, you're alternating between theta and delta modes, perhaps delta is also an important part of the encoding that's going on. And the second take-home is simply that clustering the spectrogram is a powerful way to distinguish network modes. There are many extensions of this that we're currently working on, one of which is clustering the coherence between two regions. But with that, I need to say thank you and take any questions that you might have.
Thank you, Nathan. I think we're running a little bit out of time. Just to remind everybody, you can post your question on our star, and Nathan can answer them over there. I have a very, very quick question. So all of your experiments, the data that you got are for foraging experiments, is that correct?
They're not. There's no task whatsoever. We did the parallel of all of this during a foraging task, but all of the data, just for purposes of artifacts and motivational state and that kind of thing, the data that I'm showing here is just absolutely nothing other than the rat wandering.
So my question is, do you expect anything different if the rat had to use speed for a particular reason, right? So if speed is actually informational, or if speed allows the rat to do something versus something else, in the case, especially in the case of memory, when it becomes relevant.
Right. So that's an interesting question, because normally my answer would begin that depending upon what task the animal's doing, you would certainly expect different, let's call them thoughts, that the rat may be engaged in, and therefore different modes would go along with that. But it sounds like if what you're suggesting is something different than the normal question, which is that running speed itself is informational for what the rat is doing. That would be a great instance where you might expect a combination of rhythms present, because I like to use the abstract terms here, but if the animal is running around mindlessly, then you may expect to see purely a theta mode. Whereas if the animal is thinking about something, or potentially encoding, or potentially retrieving for memory-guided behavior, or if that animal is actually having to be conscious of their running speed, in order to use that information in some way, as you were describing, then yeah, you might expect to see a different pattern in the power spectral density and different coherence between structures, because the circuit would be engaged in a different way for those cognitive tasks, as opposed to when, in this case, there is no task to be performing. So.
Great, thank you very much.
I don't know of anything of any tasks where a person has actually used the running speed as informational, however. So it's a little bit hand-wavy. So.
Yeah, great. Thanks a lot. Oh, there is one more question. Let me see if I can. All right, one question for Alexandra. What made you choose above 6 hertz, 6 to 10 hertz as a range for theta? Interesting choice. People usually choose 4 to 3 hertz.
4 to 3? Well, so
maybe
depending upon the system you talk about, and depending upon the experimenter that you talk about, 4 to 12 is one range, 5 to 10, 6 to 10. Really, everyone has titrated their own data ranges, I would say, unless somebody has a favorite, and hopefully that doesn't offend anyone. But for my purposes, the theta peak is almost always between 6 and 10. I've used 5 and 10 as well, which doesn't change anything. To distinguish from delta, it is kind of it's relevant to have a little bit of a gap between the two ranges, 1 to 4 and 6 to 10. But having played with all of that, it really doesn't matter in terms of these analyses. In some cases, I used the area under the power spectral, under the PSD. In other cases, I used the actual peak. And really nothing that you manipulate in those kinds of ways affects the qualitative result.
Thank you very much. The poster session is starting, so we need to quit here. And again, all the questions that you have, additional questions, can be posted on northstar.org. There is a link in the chat window. Thanks, Nathan. Appreciate it. And sorry about the.
Appreciate your time.
Okay. Bye-bye.
Patiently waiting for us.
Okay. Hi, Thomas.
Hello, all. I think we will wait a few more moments here to let people, if they're not paying attention to the time, they can shift from posters over to this session. So we will wait a few moments before we get started with the follow-up Q&A session from this morning's keynote. Maybe I'll put this screen up so that if people want to start reading, they can read some of the questions.
Okay. So, oh, sorry. Let me go a little bit. I just took it away. Why did I take it away? Sorry. I just. Sometimes you click the wrong thing.
Okay, I'll unfocus that screen while you find it, and then.
Okay. Yeah, I take it away. Oh, come on. Let me take this away, and then here. Oh, my God, too many screws open. Oh, not this one. Not this one. And what is it? Here, come on, where is it? Maybe it's here. Oh no. Where is it? Just click this one. Where is it? Come on. That's strange. How can it just go away? Not this one.
Hidden there somewhere.
Come on. Let me open that again. I see it in the foot. But somehow, it's interesting. How come? Maybe I can say save and then open it again. I don't want to destroy it because I did not save it. Oh, yeah.
Well, there is a record of it.
Here's a.
Oh, okay.
It's under. Oh, save. Yes, please. Oh, so cancel, and then pull it up. My goodness.
There is a video record of.
Let me save also. What is it? Oh, here is queuing it for today, and I also save it. Save. When too many things open is not a good idea, but not at least I save.
While you're doing that, I'm just going to give a little bit of intro for people joining us here. I'll just say that we have moved the questions forward that weren't answered in the first session. However, I don't see that the people who asked those questions are now flagged. And so if you'd like to identify if you have a question from before, because I'd like to invite people on screen, but as I say, we don't actually have the identities of the people who asked the questions from this morning, and they might also not be here. So if you did ask a question from this morning, please write in the chat what the nature of your question, and then that is if you'd like to ask it or have any sort of back and forth dialogue. Yeah, so Tom, I can't actually see the names. Normally you can, and for any newly asked questions, I can see the name of the question asker, but of the ones that were shifted forward from the morning session, the question asker is actually not. You can then look them up in the people list. Yeah, no, there's actually, I know where the invite user on screen and the name there is part of the question. No, it is as if OCNS is the question asker.
If you ask a question.
In all of the cases. So
interesting.
You're referring to under the start answering box.
Oh, there is one, for instance.
Okay.
There's a name.
So you can't see what I think what I'm looking at here. Where it says that OCNS. Okay. Second question, Peter Mazalik at the end of the text. Okay. Well. Okay. I see it now. And okay. So yes. And I will try to find those names. Okay, and I see some text of people that aren't able to come on. Okay, so I think what we'll do here is Anand Pathak available to ask the question. If not, then what I'm going to do is just ask it. If I don't see you pop up in chat, and then we can go. So if you do want to ask a follow-up question, then please go ahead. So now we're going to get started here. I'll ask the first question with the highest number of votes. I think these are votes maybe from just now, but the question is the salience in the fused image case possibly due to the minute jittery relative motion between the two eyes such that the bar on the right screen would appear to be slightly vibrating with respect to other bars?
That's a very good question. It's not the case because we also did a control experiment asking people to actually tell us which bar is in the other eye, and they cannot tell. So obviously they cannot see it. And they don't even have to tell us which bar. Sometimes there is no bar, so we just ask them, is there a bar at all in a different eye? Even that they cannot tell. So the jittering obviously is not enough to make them identify which the unique bar. Yeah, I think this is referring to Let me go to that slide.
So we do have the question asker with us here. If I don't know if that answers your question, or if you want to clarify any points or add something to it.
Yes, what I was asking was that in that particular experiment where the two boxes were there and the image was fused, and am I audible?
Your audio is not very good.
I'm so sorry about that. Actually, I'm logged in as a moderator also, so it's, I'm right now on the cell phone.
Okay, I think this might not work too well. But you can, for instance, if you want to add any details to the chat, I can relay them, or I think our speaker can also see the chat. Are you able, Xiaoping, are you able to see the chat as well? So
okay.
I think the audio maybe it's not gonna happen. Sorry about that, Anand.
Okay.
So I'm gonna.
But I more or less. Oh, sorry, my PowerPoint is not responding. No wonder it's. But I understand the question. Why is it echo? Can you hear echo?
I think it's okay now.
Oh, it's okay. Okay, so let me reopen this PowerPoint. Okay, and so you are worried that.
Oh, now we have the echo again.
Yeah, what is the echo? Let me take off.
Oh, can you remove that? We're not hearing you now. I think you might have to. Can you try toggling your mic? I see that it's turned off at the top of your screen. If you mouse over, I'm trying to do it, but I can't. Oh, okay. We're getting reconnection here.
Is it better?
Yes, and we can hear you.
Is it still echo?
There is a little bit of echo, but I think it may correct itself.
So how about let me take this off and retoggle.
Okay, now we can't hear you. Okay, and I got that. Okay, I can't hear you. Although you don't seem to be muted right now. Is it on your microphone? No, we don't hear you right now. Can't hear you. Okay, I see that your mic is toggled to mute right now. Can you turn the mute off? Hopefully then it will work. Can you hear me now?
Better?
Yes.
Yes.
Yes. Okay. I think there might be a concern. Do you have a second device with like a second window open? Because I think if maybe you have.
Yes, I do have a second one. Sorry.
So maybe at least you can mute that second one. Well, I guess you're not invited on screen with that. So
I just turned off the second one. Is it
okay?
better?
I think the audio is good now. everyone?
Okay,
I think so. Okay. So Anand in the chat, he wrote a follow-up question that we couldn't get with the audio. So I wanted to know whether the saliency in the vertical bar in the right box is due to inherent neural architecture or due to subtle vibration between both images.
Oh, I see. Is this when one image is in the left eye and another image in the right eye? Yeah, if that's the case, it's not due to sudden vibration, because we ask observer to tell us if they can tell whether there is a bar in a different eye, and the observers cannot do that task. That means there isn't enough vibration for them to even tell whether there is a bar in a different eye. So this is actually gaze shift to this unique bar in a unique eye without the observer even be able to tell whether it's such a unique. So it's completely dissociation between the awareness and gaze shift. So it's not because there is a movement. Yeah.
Good. Okay, so we're going to try to move on to another question here. And there is one from Peter Mazalik. Is Peter available to ask the question? If so, please comment and I'll invite you on screen. Okay, I'm going to start asking the question, and then Peter can join us if he's around. So as a follow-up to the question about the bottleneck and the amount of information we can receive, isn't it one of the insights of information theory that visual data around us is really redundant? Therefore, it could be compressed by getting rid of redundancy and using an encoding that maximizes entropy of the distribution.
Yes. And so first of all, let's say how much redundancy, how much can you compress? So we have 20 images per second coming in at 1 megabytes per image. So it's 20 megabytes per second of data coming in. That's raw data, not compressed. Okay, so how much can you compress? The JPEG and MPEG is very good at compression. They can compress by a factor of 100. So therefore, you can compress from 20 megabytes to 1 megabytes, let's say. It's this order of magnitude. And 1 megabyte is still too much. Okay, so 1 megabyte is a huge book of War and Peace is what you can put in 1 megabyte. So you cannot read in 1 second. And so if you can indeed compress them, all of them, then you will not have attentional blindness. The fact that experimental data tell you, we do not see a gorilla in our midst, we do not see things like that. That means our blindness is not trivial. Our blindness is huge. And so therefore, it's not possible for us to have such, the compression is not enough. You have to delete. And then you can say, why is it that we never fail? You know, if the data is 1 megabytes per second, and we can only read here 40 megabytes, less than 1%, how come we do not feel that we are blind? We're essentially blind because 99% of information is thrown away. And this sounds a philosophical question, but let me give you an analogy. Imagine you were born blind. You never had eyes. Would you know that you're blind? You wouldn't. Until your neighbor tells you that you are blind, you don't have eyes. Then you cannot even comprehend what your neighbor means. Okay, so we do not have such neighbor to tell us that we are blind. We are born so blind, all of us. And so that's why it's only 1999 is the first time we realized this inattentional blindness in a heavy manner. Of course, previously people kind of noticed it before, but it didn't make it such a big thing. And so it was coming as a surprise because we were born that way. Yeah, and only the experimental data tell us, oh, we did not even see gorillas in our midst. This is our neighbor. Our neighbor finally told us, then we appreciate we're blind. And so this is just making me, at least, very much appreciate how intelligent our brain is. Despite only having 40 bits per second out of 1 megabyte per second, we can still survive and not being eaten by the tiger jumping at us or by the car hitting us. That means we chose the right 40 bits out of 1 megabyte to pay our attention to, so we save our lives. That's amazing. And therefore, choosing which 2 sentences out of this book to pay attention to is even perhaps, in my opinion, more important than actually recognize, was it a tiger jumping at me or was it a cat jumping at me? Yeah. Well, cat, I wouldn't run away. Tiger, I will run away. Yeah. So.
Great. Okay. Thank you. There was a highly ranked question from this morning, so I'm going to jump down. The question was from Shivangi. If you're available, please mention this on chat. But I'm going to ask the question in the meantime. But if you mention something on chat, I'll invite you on screen as well. So the question was, what is the formation of the salience map based on?
So formation of the salience map, I'm not sure I understand, but salience map is formation in V1. It's based on the firing rate of V1 neurons. I'm not sure whether I understand the question enough to actually answer, is this the right answer or not? Yeah. First of all, the salience map is equal to the V1 firing neuron is a hypothesis. And this hypothesis then says, okay, how does it do it? Because the computational idea is there's a saliency map in V1. And then what's the algorithm? V1 takes the feed-forward elicit field, which is the Hebrew-Weasel neurons. Then it does this surround suppression using the algorithm. Surround suppression is the algorithm, and using the implementation detail as recurrent connections in V1 to make each firing rate of V1 neurons dependent not only on the visual input within its elicit field, but also input beyond in a surround, such that the visual response, V1 neural response, depends on the context. So therefore, a vertical bar is salient in the context of horizontal bars, but not salient in the context of other vertical bars. Does that make sense? Okay.
I think so. We don't have the question asker here, but hopefully that was going to answer. So there was a follow-up question to the previous question that came up in chat. So I have the question asker here with us. So please go ahead.
Great. Yeah, please.
Hopefully the audio is going to work.
I hope so as well. Is it okay?
Yes.
Yeah, that's good.
Thank you so much for answering the questions. So I keep being held up by the 40 bits per second. So I understand it is a measurement from the 50s.
Yeah.
How was this established? Was this black box measurement, or did they put probes into the connections between the V1 and the V2?
No, it's going from the connections. So if you want to measure an information channel, there's always an input to the channel, then output to the channel.
Yes.
So here the channel is a human person. The input is the image to our eyes. The output is our report. Our human person is the channel. Yeah. Yeah.
I can answer my follow-up question. Then I can answer my follow-up. So you measured 40 bits per second on these interfaces. But in your model, you say that there is only 40 bits per second going from the V1 to the V2.
No. I said, if you put vision as encoding, selection, and decoding, and so then the selection will eventually go towards 40 bits. But I say it starts at V1. So it's possible that before, so we realize it's 20 megabytes at the retina. And then it's compressed outside at the end of the, beyond the retina optic nerve, it's 1 megabyte. Okay. And 1 megabyte entering V1. And I say, okay, start the output of V1, it should start to go down because by then you already select to direct your gaze. But it goes down. It could be only go.
How do you know that there is a reduction over there? Because you're measuring at the eye. You say that you measure the encoding on the mouth and on the eye. How do you know that there is a reduction in information between the V1 and the V2?
Okay, first of all, it's a theoretical hypothesis, then it's data. Okay, so theoretical hypothesis is that since V1 is already guiding your gaze, it's already doing the selection. Computationally, we are on computational neuroscience conference, you say if it's already selecting, once it's selected, there's no point keep on the non-selected information forward unless you are full of energy, you don't mind wasting your brain resources. So it's a, first of all, logical argument. Secondly, you're right. Even if you argue that way, maybe your argument is wrong. Maybe it still keep feeding all the information. But so let's see how much from V1 to V2 it start to go down. Does it go all the way directed from 1 megabyte to 40 bits? Or perhaps it only go from 1 mega to half megabyte? Or perhaps
it goes
only
to
from
20
1 mega,
megabits.
maybe go to
It could
20
go to 100 megabits.
Oh, you
Or
mean
even
expand? Information cannot expand. Information can only go down. Okay, yeah. So therefore, it cannot go from 1 megabyte to more than 1 megabyte. Okay, at the maximum, it keep being 1 megabyte. But let's see, there's already something dropping. What information is missing in V2 that's not in V1? What information is in V1 not in is already missing in V2? Do you know?
Are there direct projections from the LGN to V2?
Maybe, yes, but
if
even
those
then,
are
in
there,
V2,
there is no missing
so
information.
Actually, from today's talk, I give you a quiz. What information is missing at V2 that's not in V1?
The one with the boxes that shifts over there, that is dependent on the 2i information.
Exactly. By V2, you already miss the I of origin information.
In your experiment?
Yeah, in the experiment, but it's also in the data. Hubert Wiesel, ever since in 1960s, they found V2 neurons are mostly binocular. And V1 neurons are monocular and binocular. So therefore, by V2, if you take V2's neuron firing, you cannot decode what's the I of origin. And so therefore, it's not just the human reporting. They cannot tell you whether this is coming from left eye or right eye. It's already missing. Okay, there's other information also missing. What's the other information? The complex cells of V1 are missing information because their firing is invariant to exact phase relationships. So if the simple cells do not feed all information to V2, mostly complex cell carry the task, then you are also missing all the spatial details. So it's actually an empirical question. I like to motivate this experimental investigation to actually quantify how much information is missing in V2 so that we can appreciate. So that's why I left it as a margin. I say, is it really all the way to 40 bits? I doubt it. But is it 0.9 megabytes? I doubt it too. I wonder maybe, go from 1 megabyte to order of magnitude half a megabyte. But even if it's half a megabyte, it's already a disaster for an electrophysiologist. Why? Imagine you're measuring a V2's resistive field. You stick the electrode into a V2 neuron, and visual information needs to presented on the screen. You measure the neurons firing, and the information is coming in. But what if this information does not reach this neuron and reaches on this trial, not that trial? Why? Because sometimes it's going to the bottleneck, sometimes not going to the bottleneck. It's not repeatable. This is why electrophysiologists always say V2 and extrastriate cortical resistive fields are more difficult to measure. Well, human weasels are lucky. They have it all. Whether it's the animals anesthetized or not, it doesn't matter. Yeah. And so in this sense, we have lots of these hints are indicating to us that from V1 to V2, information is starting to go down a leaking pipeline. And I would like to use this framework to motivate experiment to actually quantify how much leaking is leaking. Yeah.
So I think Jim had a sort of related
Hi, Jim.
question. I think you guys know each other, right?
Yeah.
I'm going to kick you off.
Jim is my teacher at Caltech. I learned lots of. Yeah, thank you. Nice to see you, Jim.
Nice to see you too. Yes, I was going to ask what it's like after many years to actually be doing experimental neuroscience, but I have a question about this subject. So a tremendous amount of work in visual system has kind of assumed that we get information, it goes in, it goes in, it goes in, it goes in. Your work and other work suggests that actually we know a lot about, or we assume a lot about the world just in how we point our eyes, right? So that implies that there's actually an internal model that we're working with as we seek information, right? Which explains why some people see the world that I don't see, okay? Like prominent people in the United States. But anyway, so my question is this from the point of view of information, okay? If what happens in an information sense If you have a sophisticated internal model of the world, which is what you're using to organize your seeking of information, okay, then what happens in an information sense going from V1 to V2 if what you're now doing, for example, is interacting with and doing something which is in relation to this internal model? So the idea of, sort of leaking information, that the further you go in, you have lower and lower amounts of information. It could be in a larger sense, sort of a cognitive sense. Isn't it possible that information from the outside going in, when combined with the information that's already there, in effect produces a larger, deeper amount of information or context for behavior? Do you see what I'm saying?
Yes, you cannot create more information, but inside your brain, what you have is actually a code book. Okay, so you have a code entering, then you need a code book to kind of look up. So when I say that you have a feedback in the central visual field, is that using this code book to say, hey, are these two dots really vertically lined up or horizontally lined up? This code book has the knowledge that this neuron is tuned, it's like a label line, this neuron is tuned to horizontal. And so therefore, if this neuron is firing, it must be because something, this and that. So it has this code book and say, now, because this neuron tell me this and other neuron not tell me, so therefore I wonder what are possible sets of input. And the internal knowledge knows that this neuron already restrict to me. It's not Jim Bauer in the input. It must be Steve Prescott. You know what I mean? So this is because the code book it has. And so therefore it can top-down feedback and say, I don't need to look for American, I just look for a Canadian. So when the input is ambiguous, it's like an ambiguous perception. You have this Necker cube perception. It can only be this way or another way. So it will only top-down query only A or B. It's not going to query C or D or E or F, because by the internal model, it knows it's not possible C, D. It's only possible A and B. However, whether it's A or B, internally you cannot create. It only says it's only possible A and B. Then I need to go back to V1 and still externally to query, is it really A or B? So this is what I mean. It doesn't create information. It clarify by querying for more information. Yeah. And this is why if you have a visual input and suddenly mask it, okay, you present the input so short that you mask, the backward masking can create problems, because when it's going back to query, you give us some other input, then this is, so we predict that this backward masking is more severe in problematic in central visual field, because the feedback is in central visual field. So a lot of these framework can I
understand it knows how to interpret the code based on what the neurons do and don't do. My question is, and it's a hard thing to get at, in the olfactory system, we think that, I suspect that the olfactory cortex has a map of the metabolic world is accumulated over evolutionary time. So when the olfactory system is actually, to go back to a subject that you used to think about. Yes. When the olfactory system is actually doing a recognition, it's getting information, but that's referenced to this deep knowledge that it has about the olfactory world. And so really understanding how it works, you need to understand something about the reference frame that's provided by this deep historical evolutionary model. And I know people in vision know, play around with that, right? We know that bright things are usually up and dark things are usually down. I think it's a hard problem, but it's an interesting problem to ask, which sort of closes the loop that you're talking about. How sophisticated and how much knowledge does the brain actually have? Okay. That it is using as a reference for the data that's coming in. So,
this is very interesting. In fact, my work on vision is a lot inspired by my olfactory work, which I did in the Caltech as my PhD thesis. You were there as well. I was working with John Hopfield. And so one of these things is indeed olfactory cortex feedback to the olfactory bulb. It causes, this is the hypothesis in the thesis, that this top-down feedback actually cancel out the background to do the foreground recognition. And this is in fact trying to meet the expectation, explain away the background, get the foreground. And it can cause problems, just like backward masking. What happens is if I smell my dog in my kitchen, yeah, very good. Then on top of that, you have the coffee. And then you can subtract the background dog and you get the foreground coffee. But what if when I'm presented the coffee at the same time, within the same sniff, take away the dog you just get subtracted from? Now I will sniff coffee will smell different because I thought the dog was still there. This is called cross-adaptation because you just fool the system. Yes, this top-down feedback, whether it's an olfactory system, it's a backward masking, meta-contrast masking, they are all kind of connected. And this is in a computational terms called analysis by synthesis, and it has to be respectful with certain rules. These rules, if you violate, you get these strange illusions or problems. They can become diagnostic for the underlying computational algorithm. Yeah, very.
So everyone should learn about the olfactory system before they go anywhere close to vision. Otherwise, you're lost. Okay, thank you very much.
Thank you. Yeah, thank you.
We do have a quick follow-up question that maybe you could address quickly before we move on to the next question. And that was from the previous question asker. Can an input result in memories being recalled, adding to the information content of an experience? I think you partly answered that, but do you want to address that question directly?
Can an input result in memory being recalled as adding the information content of an experience? Actually, I do not completely understand the question very well. In a sense that maybe I'll use an olfactory example. Okay, so my colleague at earlier in UCL, in fact, I was discussing with him on actually Jay Goodnot. Is it Goodnot? Yeah. So they have this experiment where they ask the subject, is there something smell there? Okay, they give the sniff some smell, and it's so weak the subject cannot tell very well. And then at the same time, if you show them an image, let's say you show them an image of an orange, then they are more likely, imagine if they're sniffing orange, they are more likely to say yes in the orange. But then it's a false alarm, right? And no false alarm. Okay, false alarm rate does not go up. So therefore, having the visual image of an orange make you imagine that could be an orange. This obviously is internal knowledge, and then that I don't know whether this indirectly asked your question, but if you can please make a more specific phrasing of your question so I can answer more directly.
Okay, so I think at this point we will maybe move on. Yeah, Jim has got a comment there, but I think we'll move on to another question here because Shavika has been very patient. I'm going to invite them on screen now to ask a question that was posed in the question box before. Shavika, I've invited you. Oh, accepted and connecting. Okay, so be on here in a few moments. Just wait for it.
Maybe I can have a preview.
I'll ask the question while they're coming online. If there is an image in which one bar has different orientation than others, and there is another bar which is continuously moving, how does the V1 decide which one to focus attention on first? And we have
I see.
The question asker with us now. I don't know if you want to add anything to the question.
I see. How does V1 decide? Yeah.
How does V1 decide?
Yeah.
Nope. Just wait. We're not. Your audio is a little bit jumpy.
Okay. Okay.
So try now.
The V1 does not. V1 decides by the firing rate. So if the firing rate to a uniquely oriented bar is higher than the firing rate to the uniquely moving bar, then this is the hypothesis that higher the firing rate, the higher the saliency. And this hypothesis is confirmed in our monkey experiment with my colleague Wu Li and Yang Ying. They show that the higher the firing of V1 neuron, the faster the eye saccade to it. Yeah, so that's the hypothesis, and this hypothesis is having its prediction confirmed in the behaving monkey in the experiment. Yeah. Mm-hmm.
Does that answer your question? Oh, okay.
Yeah, so I wanted to add on a bit. So from the slides where you showed the bars of different orientations, in which you have bars of the same orientation and one bar of different orientation in between. So there, the attention, the selectivity of V1 depends on the firing rate of the corresponding neurons. So as you already said that the like neurons will suppress each other, and there is one neuron corresponding to the bar of different orientation whose firing rate will be the highest. But for the case when we have, say suppose we have an image in which we have one bar of different orientation and there is another bar which is moving, and the rest all other bars have the same orientation, then how does the V1 decide that which one is the bar in which it has to focus attention on?
I thought I already answered that question, so I answered again. V1 decides by the firing rate. So if the firing rate to one bar is higher than the firing rate to the other bar, then the firing rate with the higher firing rate is the more salient. Of course, this decision is made by a recurrent circuit in V1 where they have this like-to-like suppression. And then you can say, how does the like-to-like suppression between orientation stronger or between less stronger? So through experience, maybe through the evolution as well as through this individual animals growing up experience, it has already built up this plasticity, such that it make it this way. Of course, there's also individual differences. Maybe your V1 is slightly different from mine, such that maybe you will weigh up the orientation slightly more than me, or slightly, but qualitatively, it looks like many people are very similar, even though your V1, there's up to a size of 2 difference between one human's V1 and another human's V1. And some people, for instance, they also have strong color sensation. Some color is stronger to some people than others. So there is quite individual difference. And in my lab, in our experimental data, we find these difference. Yeah. But nevertheless, in principle, yeah, you can have the V1 decides by the firing rate.
Good. Okay, thank you. Okay, there's a couple question askers that aren't available, so I'm going to ask one of the questions. And if Grace is here, please send a chat. Is their original paper on? Okay. Sorry, in their original paper on saliency maps in the brain, Koch and Ullman, 1985, suggests LGN as where the selection first occurs, but you say V1. Why do you think LGN doesn't have this selection?
Oh, very good question. Yeah, and so first of all, if you are selecting it, then it's better that you will have your saliency map be read by somebody, read it and execute it. Eye movement shift. LGN does not project to the superior colliculus, which will read the saliency map and do the gaze shift. So it can't be the LGN. That's the main answer. I can give you secondary reasons, but this will be maybe.
Hopefully that will satisfy.
Unless somebody asks for more reasons, yeah. Okay. But that's the main reason.
No, I think that's fine. Thank you. Okay, so I'm going to read another question here, and then I'll try to find some other question askers. Is the speed and accuracy of the selection process the same for all tested specimen? I'm not sure exactly what specimen
maybe species. Yes.
Species,
yeah. Speed and accuracy trade-off is not even the same within a single animal, because we will do the trade-off. Sometimes it's more important for us to react quickly, but not accurately. Sometimes it will be more accurate and be slow, depending on the context. So even individual animals will change that trade-off. So not to mention between animals, yeah.
Right, and I think you also referred to the central versus peripheral differences between species earlier and whatnot. Okay.
Yeah.
Thanks. Well, okay, we'll do that one. Okay, so there's a question from Alex. I'm going to read it out. Recent work has highlighted the role of disinhibitory V1 cortical VIP neurons in poking holes in the blanket of inhibition. That's in quotes. That is inhibition of inhibitory interneurons, which could contribute to generating a spotlight of attention. Given what is known about decoding of visual information across visual cortical areas, is it likely that there are similar or very different local neural mechanisms processing information across different areas?
Yes. So for instance, in my proposed framework, you have encoding, selection, and decoding. So if V1 is doing the selection, let's say V1 is not doing decoding at all. In fact, I think V1 is doing decoding, as I say, there's feedforward feedback. So some other area is doing decoding and not doing saliency. So it must be different. And the data so far shows that at least bottom-up saliency is almost exclusively in V1. Of course, we can find more data to say maybe it's not exactly exclusively, but V1 is dominantly computing bottom-up saliency. That means other visual areas cannot be doing that so much. Then by definition, if that's the case, they cannot be the same. Yeah.
Okay. I'm just looking here to see if Wouter would like to ask his next question. I will start it, and then he can join us on stage if there's any follow-up. So the V1 SH saliency or saliency hypothesis mentions a firing rate as determining factor. I think this came up in an earlier answer. To assess a rate of neuron you need to integrate over time, could the saliency be encoded into a sparse parallel coding? This might be faster and match better with the most reflex-like behavior of eye gaze.
Oh, I see. Very good question. Yes. So even though firing rate is a determining factor, but in fact, equivalent is just the first spike latency. Okay. So one very inaccurate way, but it's very quick way. This is another speed accuracy trade-off. Is just say first spike coming from whom? First spike coming from a neuron in the upper visual field, this location, then that's more salient. Okay. Then the latency to the first spike is the quickest way.
Triggered by a saccade or something like that? I mean, what's the reference to that latency?
So let's say there is because it can be from the last saccade. So let's say that from the last saccade, you fixate on something, and then you investigate and decode and say, hey, it's a face, somebody. And then you want to go to the next one. It could be counted from the last saccadic landing, or counted from somehow you're done with this task, you're ready to go. I think you can formulate a different way if you like. But in principle, yes, you're right. The longer you can wait to count the more spikes, the maybe better accurate. But sometimes you don't need to be accurate. You just want to act fast. So if a tiger is jumping at you, you don't want to say, is this a male tiger or female tiger? Who cares? Just run. Yeah, you don't have to count how many teeth this tiger has. Just go. Yeah. So you don't have to wait for the second spike. Just run.
Okay. Thank you. Okay. We're coming close to the end of the questions, and I think Ben had suggested that his internet connection would preclude him from coming on. So I'm going to ask the question: how does this new framework compare to predictive coding / processing framework that proposes the predicted stimuli are suppressed from further processing and only unpredicted stimuli registers get attended to?
Predictive coding, okay. I have a framework which talk about encoding and selection and decoding. In encoding, usually people talk about Shannon information encoding. That means you just talk about information bits and you don't talk about content. In that, you already have predictive coding. Predictive coding, this framework has been around since even information theory days, yeah? And so, do you mean that predictive coding? Yeah. And on the selection bit, there is top-down selection and bottom-up selection. A lot of the top-down selection is predicted selection. So I will for instance, look towards the doorway because I predict my dog will walk in that way. Yeah, and so I will, in anticipation, your anticipation is as prediction. On the other hand, because my dog is walking that way, I don't want to look into it. And so there's lots of prediction. Do you mean that? Or the predicted coding is not only in selection, could be in decoding. In decoding, then when I say you want to feedback and query to say whether input is A or B, a lot of it is your internal knowledge. What do you mean? And I can better answer your question if you can more precisely put it, because otherwise it's very difficult. You can say it's too general. You can say it's saliency map out of the evolution, and then yeah, sure, and that's a too general question.
Yeah, so I don't know if Ben is available to pose a follow-up here, but isn't that predicted? So maybe part of the specific detail here is our predicted stimuli suppressed? I mean, so are they almost like, so you're selecting the most the unpredicted?
So let's say in encoding,
is part of that is selection part of this predictive coding, is the prior somehow represented in what is selected? I mean, maybe that's part of the question.
Oh, okay. So let's focus on the selection bit then. So things that pop out, for instance, vertical bar out of horizontal bar that pop out is the most unpredicted part. Okay, if you look at the context, everything is horizontal. And then you just cover the vertical. Then you ask people, what's behind my thumb? I just cover that vertical bar. People guess, is it horizontal, vertical? You will guess, predict it horizontal. And you raise the occluder, say, oh, wow, vertical. That is the most unpredicted part. And that used to be, that can be the most salient. So sometimes people equate saliency as surprise. You did not predict. It's just so that the surprise works 100%, not 100% of the time. Sometimes things unpredicted, it's not salient. And so therefore, surprise is not 100% correct measurement of saliency. Operation measures saliency and how strong is attraction. But anyway, just for prediction, saliency is the most unpredicted part. But top-down selection, sometimes you will use your prediction. Yeah. And so predicted coding, do you mean encoding or decoding? Okay, selection is not coding.
I'm not sure.
Yeah.
Okay, so and Ben is not here to ask a follow-up. So okay, we do have 2 questions left, and I think we're going to
here.
leave I'm going to invite Thomas on because we'll leave Jim's very general question as perhaps the last one to end the session. So Thomas should be joining us. And his question, I'll just read it off, and then no, he's here. Go ahead, Thomas.
Oh, hi, Thomas. Hi, Thomas.
Yeah, it's a bit irritating, this little delay, isn't it? I just was wondering, it seemed to me that your optical illusions depend a lot on the shape of the receptor fields of the early neurons in the system, so that are bore-like with a plus and a minus, right? Is it just inevitable that receptor fields look like that, or is the visual illusion just very specific to how the human system works?
I wonder. But because in cats, they also have these receptor fields. Of course, we know in V1 we have bar detectors and edge detectors. In the morning, I kind of used the edge detector. It worked. So I suspect if you have an animal that only has bar detector, maybe it would not work. So that would be something interesting. Can we find such an animal? And then see this illusion doesn't work on it. Yeah. And
to the vulnerability of feed-forward artificial neural networks. So there, of course, you can manipulate what kind of filters they have.
That's right. Yeah. In this artificial neural network, you can present a panda and then you change a little bit of the texture. It's a given. A lot of these changing structures are really at these pixel level. Just play with the texture of the V1 with the field and V2 with the field, and somehow they learn it this way, and they can just fool the system if they don't have the feedback to come back and say. For instance, when you come back and say, "Oh, it's a panda," then panda should have 2 eyes, or black here and then something like that. And if you have this top-down feedback, then you will not be fooled by it. And the reason these adversarial attacks so successful on this network is they don't have this top-down feedback. In a sense, when the generative model says, if it's a conditional probability, what should be my pixels on the image, given conditional on that it's a panda, then it will not be full. So in a sense, there's this top-down feedback. When I say it's the computation of analysis by synthesis, is I'm trying to analyze the feed-forward pixels by synthesizing what these pixels should be if it's a panda. Yeah. And then you can compare, is it really a panda? Oh God, it's not a panda. I won't be fooled. Or is it really giving? It's not a fool. And this is in a sense that, in that example I used, Mary neuron is firing. And I say, okay, Mary neuron, I know it's two Gabor patches in the on-field and off-field. Then I say, what could be, you can predict. If it's firing, then I predict that it could be two white dots lined up horizontally, two black dots, it could be this, it could be that. And so it feedback with all these kind of likelihood functions. It's like a given Mary neurons firing, what is the likely input? It's a conditional probability. And then within that conditional probability distribution of likely input, you say, is it A or B or C or D? So imagine indeed you only have 4 choices. Then it's only 2 bits of information to uniquely nail down which one of these 4 choices is. And so that means in this way, the top-down feedback is querying from V1. Give me two additional bits of information, specifically only on this. So if the information bottleneck is 40 bits per second, then asking for 2 additional bits in the next second is not too much of a luxury. It's okay, it's still allowable. However, if you ask a general question, is this a beach? Is this a mountain? There's a million possibility, then it will be too many bits. It cannot. And so in this sense that you feed forward giving heuristics of just a few possibilities, then feedback to narrow it down with your internal model of what this Mary neuron is telling you about. Yeah.
Very cool illusions, by the way. I really enjoyed those. That was
Thank
great.
you very much. Yes. Thank you.
Good.
Yeah.
Okay, I'm going to kick you off, Thomas. Bye. Okay, we have one final question here, and it was actually posed by Jim. And the question was, what is it like returning to run an experimental lab after all these years? And maybe I can just expand that a little bit and say, can you maybe comment, provide advice to people looking at how to combine experiments, with with more theory and what your experience is, over the years of making that combination work.
Yes, I thank you, Jim, for this question. I like to advocate that modern day biology students should not fear that they can learn a quantitative method. They can be very good theorists because they know biology. Or another way, I have a slogan. My slogan is, the best theorist is an experimentalist. The other way around is, the best experimentalist is a theorist. And so I offend nobody, but I'm making everyone feel that you are able to do that. A natural scientist, a natural science, we make hypotheses, we observe nature, and then we extract regularities in nature and make them laws. And then we say, is this only a hypothesis? Go back to nature to test and falsify laws and get better ones. And we can make progress if we can do this well. Doing this well requires collaborations with people of different expertise, and I'm very lucky to have collaborated a lot with experimentalists. And I actually learned initial physiology from Jim Bauer's Jim's course, and I did my rat electrophysiology surgery learning in Jim Bauer's lab. And that really made me appreciate doing experiments. And I learned a lot from my current colleagues like Uli, my collaborator, and he's such an expert on monkey electrophysiology, and he has great interest in primary visual cortex and top-down attention. I'm interested in bottom-up attention. I learned a lot from him, and so I appreciate a lot of these things. And there is a sense of fear in the beginning. I feel like, I'm a theorist, am I good at doing experiment? But the more I learn from experiment people, the more I can gain confidence. And I'm sure it's the same with the experimental people, once you venture into computation, you will gain confidence, and that I think that's the future. And I like to encourage everybody to try that.
Great.
Mm-hmm.
Okay, I think we are getting very close to the end of our hour, and I think this is a good place to finish off with a sort of aspirational note. And so if there are any additional questions, I think they can be posed on Neurostars. But this is the end of our session now. This session will be, it is recorded, so it can still be watched again afterwards if you want to go back to it. And with that, again, I'd like to thank our speaker. I think that was a great session this morning. I think people really enjoyed the illusions, and I think this is a very helpful question period as well as a follow-up. So thank you again. And I know there is a party that is to get started here very shortly, so we should adjourn and everyone enjoy the party. And thank you again to Xiaoping.
Thank you.
Good night, everyone.
Mm-hmm.